grounds. Number one, he should know more than the speaker about subject. If not, he should be a senior man in age. Maybe the second criteria made me to chair the session. Anyway, now I request Professor Benedict G. Machado, Department of Physiology, School of Medicine, Ribeirão Preto, University of Sao Paulo, Brazil, to come to the stage and deliver his lecture. Welcome, sir.
Good morning. Thank you for the introduction. And I'm wish to say good morning to everyone. And always is a pleasure to be in India, and especially in this meeting of APPI, which is really a great meeting. I have a great time interact with my colleagues, physiologists here in India. And as I said yesterday, I hope that this is just the beginning of several new collaborations among the physiologists of our nations, since more recently Brazil and India are interacting in different areas of science and technology. And we hope that physiology will be one of these areas that we can take advantage if I can help in any way in strengthen this cooperation in between India and Brazil. I will be more than happy. Okay, today I will be present some of our current projects in the laboratory, some data that we are not yet published. That is for us is a new approach to understand the neuron astrocytes interaction in the brainstem of mice exposed to hypoxia. I work with hypoxia for several years. Most of my studies were in rats, and recently, due to the genetics models of mice, we moved to mice, but mice were a big challenge for us. And I believe that is for all the researchers. Mice are very complex experimental models. You need to take care when you perform experimental physiology in mice. So this is my college in the city of Ribeirão Preto, 300 kilometers north to the capital. That is the city of São Paulo. This is our college of medicine. This is the hospital, the kids' hospital, and this is the central, the basic research building. My laboratory is around here in the second floor. Okay, let's start where I can move. This one is here. Okay. I will talk mainly about the nucleus stratus solitarius, which is an important region in the dorsal aspect of the brainstem, which integrates the neuronal inputs from peripheral chemoreceptors. So in this cartoon, I am illustrating in case of lowering of the partial pressure of oxygen in arterial bloods, the carotid body are activated, sending projections to the NTS, which is the first synaptic station in the brainstem before higher order neurons connected with the ventral aspects of the medulla, where are generated and modulated the respiratory as well the autonomic activity to the cardiovascular system. In my laboratory, we are exploring NTS as well these ventral aspects of the medulla using different experimental approaches. When these peripheral chemoreceptors are activated, usually they produce increasing sympathetic activity, vasoconstriction, increasing the parasympathetic tone to the hearts, reducing the heart rates, as well producing the expected increase in the respiratory frequency. So today my focus is the NTS, to understand the changes that occur at the synaptic level in the NTS of mice exposed to sustained hypoxia. So in previous studies, as I mentioned before, performed in rats, sustained hypoxia increases neuronal excitability and glutamatergic transmission in the NTS neurons of rats, which may contribute to the sympathetic hyperactivity and hypertension. For several years, we used this model of chronic intermittent hypoxia and sustained hypoxia in rats because it produced sympathetic hyperactivity and hypertension. This was an experimental model of neurogenic hypertension for us. When we changed the model to mice and exposed these rodents to the same experimental protocol of sustained hypoxia, we observe quite a different response. Sustained hypoxia in mice increases the parasympathetic tone to the hearts and the pulmonary ventilation, but produce no changes in the sympathetic activity. So the pattern of the physiological response to sustained hypoxia in mice are quite different when compared to rats. This was a first surprise for us. We were not expecting this when we start to work with mice. But this is the nature. We must understand the experimental models that we are using. And we are learning everything again with mice because they are different. So the focus of our experiments that I will be presenting today is the so-called tripartite excitatory synapses in the nucleus stratus solitarius. In this cartoon, I am representing one of synaptic transmission in the NTS, the presynaptic, the postsynaptic aspects, and also the astrocytes, which involve part of the synapses in the NTS. So our idea was to explore what are the changes since these mice also present increase in the excitability of NTS neurons, if these changes are due to changes at the presynaptic or postsynaptic, or even with the contribution of changes in the astrocytes modulation of this synaptic transmission. So the tripartite synapses is a functional interaction between neuronal terminals and astrocyte processes. It's not the body of astrocytes, but really the process of the astrocytes that interact at synaptic level. The neuronal activity can be modulated, as I said, by pre and postsynaptic and astrocyte mechanisms. And astrocytes modulate glutamate concentration in the synaptic clefts via glutamate transporters, GLT1 and GLASTs. With these transporters, they remove the glutamate from the synaptic cleft to avoid any damage to the postsynaptic neurons and to avoid hyperexcitability. So the glutamatergic synaptic transmission, we look to the AMPA and NMDA currents in the NTS of NTS neurons of mice exposed to sustained hypoxia. In this cartoon, I am presenting here the micropipettes that will be recording the electrophysiological activity of these postsynaptic neurons. And here the stimulation of electrodes to depolarize this postsynaptic neuron. And here the focus is the postsynaptic aspects of the synaptic transmission. And when we look to the AMPA currents of these neurons of mice exposed to sustained hypoxia, we observe a large increase in the amplitudes of the currents of AMPA which in the group you can see a significant increase in the amplitudes of these AMPA currents. The same was observed in relation to the NMDA currents. Just remembering that the glutamates activate the ionotropic receptors, AMPA and NMDA receptors, which when activated generated these currents. So sustained hypoxia produced an increase in AMPA and NMDA currents in these mice, in the NTS neurons of mice exposed to sustained hypoxia. So sustained hypoxia increases the glutamatergic neurotransmission in the NTS of mice. So the aim of these studies that I will be presenting today were to investigate the mechanisms of increased excitatory transmission in the NTS neurons of mice exposed to sustained hypoxia. And we looked for these three different aspects: presynaptic, astrocytic modulation, and postsynaptic parameters. For this, we used these mice of this strain C57 black 6J. And these animals were maintained in the control group in normoxia for 24 hours. And the sustained hypoxia, these animals were in the chamber, hypoxic chamber, for 24 hours in 10% of the fraction of inspired oxygen. For this kind of study, we anesthetized the animals, remove the brainstem, prepare slices of brainstem containing the NETS. And most of these experiments were electrophysiological studies using whole cell patch clamp. Here is illustrative of the pipettes touching one of these NETS neurons. We also use a multi-electrodes array to cover the entire extension of the NETS. This is, in fact, the patch clamp, of course, is an intracellular recording, and the multi-electrode array is an extracellular recording to give us an idea of the overall activity of NETS neurons. We also use the calcium leaving image of this, especially the astrocytes in the NETS. As well, we evaluate the morphological characteristics of astrocytes in the control and the group of animals submitted to sustained hypoxia. So the first thing was the evaluation of excitability of NETS neurons using patch clamp. Here we, again, the recording of the postsynaptic neurons, a stimulation of the TS, the tratus solitarius, which corresponds to activation of the presynaptic neurons. And here we evaluate the action potential induced by positive injected currents. Since it's patch clamp, you can induce injected currents in these cells and induce the polarization. And you can see, comparing one cell of the NETS neurons of normoxia and sustained hypoxia, the number of action potential in response to the injection of the same currents is much, much higher. So here we have the injected currents, and here we have the number of action potential. And we can see with a large number of cells recorded that the NETS neurons of animals submitted or exposed to sustained hypoxia present a significant increase. The same occur when we induced the action potential by electrical stimulation of this by this electrode. So you can see when in one neurons of the NETS, we activate the electrical stimulation, one action potential. If you look to the cells from animals submitted to sustained hypoxia, a large train of action potentials. So if you compare this response to the train of 5 tratos solitários stimulation, we can see that the increase in the number of action potential is significantly higher in the NTS neurons of those animals exposed to sustained hypoxia. Therefore, sustained hypoxia increases excitability in the NTS neurons of mice. With respect to excitability of NTS, uh, the entire NTS of mice using the multi-array microelectrodes, here is the position. This is a dorsal view of the brainstem. This is just for orientation, the entire nucleus stratus solitarius. And here are the electrodes displayed in this area that cover the NTS. And this multi-electrode array, each electrode required, of course, more than one cell. But the waveforms, the analysis of the waveforms allowed, this is example of the one neuron, different neurons recorded by the same electrode. But we can, with this kind of analysis, identify the overall activity of NTS neurons. And then we verify that the number of events in the NTS neurons of animals exposed to sustained hypoxia was significantly increased. So it's a large number of neurons that we were able to evaluate in this case. Therefore, sustained hypoxia increases extracellular activity in the NTS neurons of mice. Excuse me. So the next step was to look first to the presynaptic parameters, as I mentioned before. So in this case, we use several theoretical ways to evaluate the activity of the presynaptic neurons. And these parameters involve mainly the probability of release of the neurotransmitters. And in this case, we use several protocols. This is the covariance of the AMPA currents, which allow us to have an estimation of the release of the neurotransmitters. And this is also explored using the paired pulse ratio, in which we apply a train of stimuli and evaluate the amplitudes of of each of these stimulation, and we compare among the groups. As well, the amplitudes of the currents when we stimulate the tratus solitarius and evaluate the excitatory postsynaptic currents. And we verify with all these kind of analysis, looking specifically to the presynaptic level, that there are no changes in any of these parameters. Therefore, sustained hypoxia does not alter presynaptic parameters in the NTS neurons of mice. So that change in the excitability that I described before, it is not due to changes in the presynaptic terminals. So the next was to analyze the modulation of astrocytes. Look that now our pipettes, micropipettes, move to the astrocytes. And we explore all in different way the role of astrocytes in the modulation of synaptic transmission. And our working hypothesis was that astrocytes should be playing a major role in the changes of this increase in the excitability. Our hypothesis was that the glutamate transporters activity should be reduced. But I will show you that we were wrong. Our hypothesis was not confirmed. So looking to the astrocytes, we identify these cells, we label the astrocytes with sulforhodamine, here in reds, and then we patch these cells. And we explore the electrophysiological properties of astrocytes. As you know, astrocytes are not excitable cells. But when we injected currents, we can depolarize these cells. It is dependent, of course, the injection of currents. And this is an example. If you increase the currents, like here, we can see change in the membrane potential. It's not the action potential because these cells are not excitable. But we verified that the membrane properties, the resistance of these membranes of astrocytes of mice exposed to sustained hypoxia was similar to that observed in the normoxia group. Therefore, sustained hypoxia does not alter the resting membrane potential of NTS astrocytes. So this parameter of astrocytes were not affected by hypoxia, sustained hypoxia. So then we start to evaluate the glutamate transporters, in fact, activity in the astrocytes. For this, the protocol, we block all the excitatory amino acid receptors of glutamates using DNQX, AP5 to block AMPA and NMDA receptors, as well the tetrodotoxin to block the activity of presynaptic neurons. And then we start to explore exclusively the activity of these glutamate transporters in astrocytes without any influence of pre or postsynaptic neurons. And to activate the glutamate transporters, we use the aspartates, which is a neutral activator of these transporters because we cannot use glutamate or any of these selective antagonists to activate the glutamate transporters because these experiments takes a long time, and these, of course, will damage not only the astrocytes, but also the postsynaptic neurons. And with this D-aspartate, you can see this is the recording of the currents of these astrocytes. And we evaluate the currents generated by the activation of this glutamate transporter in control, normoxia, as well in the group of NTS astrocytes of animals exposed to sustained hypoxia. And we, for our surprise, but for this reason that we ran experiments to test our hypothesis, and it was wrong. There is no major change in the glutamate transporters in the astrocytes in the NTS of mice because there is no significant difference. And look, always we are working with a very large number of cells. Therefore, sustained hypoxia does not alter the glutamate transporter activities in NTS astrocytes. So the next was to evaluate the changes in intracellular calcium in astrocytes. For this, we identify the astrocytes with sulforhodamine, and we use fluorophore as a marker for the intracellular calcium mobilization. And we use AMPA to activate the maximal response. So in this case, we evaluated the number of regions of interest in the areas in the NTS. This is perfusion with artificial cerebral spinal fluids, and this with AMPA. And we compare the fluorophore intensity indicating the calcium, intracellular calcium mobilization, comparing with the control. And we verified that there is no change in the intracellular calcium mobilization. Therefore, sustained hypoxia does not alter intracellular calcium mobilization in the NTS astrocytes. We also evaluate the morphology of these astrocytes using immunohistochemistry. This is a control, and this is NTS of animals submitted to sustained hypoxia, and evaluate the area of GFAP, green fluorescent protein, with indication of astrocytes, and no difference was observed. We also used transgenic animals expressing GFAP in the NTS neuron astrocytes, NTS astrocytes, and we verified that the density of GFAP, the cell process that we were able using multiphoton microscopy, the cell process, as well the cell axis of these astrocytes, they presented no change when compared with the control group. So sustained hypoxia does not modify NTS astrocytes morphology. So we explore the presynaptic, no change. We explore astrocyte modulation, no change. Let's explore the postsynaptic parameters. Now we look to these postsynaptic neurons, and the pipettes of the patch is back to the postsynaptic neuron. And then when we explore the AMPA currents, this is the protocol in which we use tetrodotoxin to block the neuronal activity, as well fluoracetates in order to block the activity of astrocytes. They are metabolic inhibitors of astrocytes. And then we apply AMPA agonists in the bats in this perfusion. And then we observe, this is the normoxia, typical example, and this is in postsynaptic neuron of NTS neurons of mice exposed to sustained hypoxia. And here we observe a large and significant increase in the current density of AMPA currents in the postsynaptic neurons of NTS of these mice. And the same was observed with respect to the NMDA currents. When we apply NMDA using similar protocol in different cells, we explore the NMDA, apply NMDA, and you can see that the amplitudes of NMDA currents was again similarly to AMPA, similarly increased in a significant way. So sustained hypoxia increased AMPA and NMDA postsynaptic currents in the NTS neurons. So that increase in the excitability of NTS neurons, in accordance with our data, this increased excitability is not due to change in the presynaptic, is not due to change in the astrocytic modulation, but it is related to change in the AMPA and NMDA postsynaptic currents. This is quite different than our previous data obtained in rats, which we published several studies. And as I said, this transition from one model of rodents, rats, to another, mice, was not a simple task. We spent much more time than we were planning in order to understand how, as is this case, the difference among rats and mice. So in summary, I showed you that sustained hypoxia increases the excitatory synaptic transmission and neuronal excitability in the NTS neurons of mice. And this change is due to increased in AMPA and NMDA postsynaptic currents, other than presynaptic parameters or astrocytes-related mechanisms. Of course, that these projects, we are running this project these days in the laboratory. And of course, this kind of data brought new questions. So what are the mechanisms underlying these changes in the increase in the AMPA and NMDA currents in these postsynaptic neurons? But for this, we reach another step in terms of methods and technologies to evaluate what are, in fact, these mechanisms. And the perspectives in this project that we are running these days is that the mechanisms underlying the increased AMPA and NMDA postsynaptic currents involving the density. This is one thing that we must explore, the density of postsynaptic receptors, because with this period of exposition to sustained hypoxia of 24 hours, we may have several important changes in the expression of the receptors in the surface of these cells, which may explain these increased excitabilities, or the kinetics of these receptors that may be affected by hypoxia. And of course, all these important issues requires another series of experiments. But for a while, our view about these changes in NTS neurons of mice, that these data are different of those previously published by ourselves in rats. But this is consistent. This difference in terms of electrophysiological properties are consistent with the physiological responses in terms of cardiovascular and respiratory responses to hypoxia in rats and mice. So the observed changes in the modulation of NTS synaptic transmission in response to sustained hypoxia in mice are different in relation to those previously described in rats. The increase in postsynaptic glutamatergic currents in the NTS of mice, as I mentioned before, is consistent with the autonomic and respiratory profile observed in these rodents after sustained hypoxia. And this is something that, although this model of experimental model of mice was not exactly what we were looking for or expecting from these rodents, we are learning a lot physiology with these little rodents. And this is a challenge not only for us, as I mentioned before, but for all laboratories that are now moving to the mice for genetic possibilities to explore all the transgenic mice. But we must be aware that their physiology is very special. Not only the mice as a unique model, but each strain of mice responds in a different way to hypoxia. This is another issue that we are involved in these days because their genetic backgrounds are quite different. So using C57 black 6J as I use in these experiments, or if we use bulb C does not strain of mice, or Swiss, third uh, strain of mice, the results in terms of the response to hypoxia are quite different because their background in the nature is different. Some of these strains came from the high altitudes, the highlanders, and some of these strains came from the lowlanders. So we cannot compare even among strains of mice. In other words, it's easier to work with rats than mice. With this, I just wish to finish the acknowledgements to my colleagues in my laboratories at the University of São Paulo in Ribeirão Preto, postdocs and PhD students, Daniela, Davi, Juliana, Carla, Ludmila, Luiz Henrique, and Mikael, a colleague at the University of Alfenas in Brazil, Silvia, and some collaboration with Professor David Klein and the postdoc Procopio at the University of Missouri in the United States. And of course, the financial support, I finish with acknowledgement to the financial support from our state foundation and the federal agencies in Brazil that make possible all these kind of studies in our laboratory. And this is a Brazilian national tree that was flourishing the last 2 weeks ago when I left our campus. This is in front of our building. This is the national Brazilian tree, IP. And with this tree, I finish my talk. And thank you very much for your attention. If we have time, I will be happy to answer questions. If not, after I'll be available all the time of the meeting. Thank you very much for your attention.
Thank you, Professor Puchado, for the wonderful in-depth talk on delineating postsynaptic responses and not the astrocyte and not presynaptic in mediation of sustained hypoxia response. So this is open for the questions and comment from the audience. Yeah, Dr. Lakshmi, please.
Please.
Somebody can give mic.
Do you have some microphone here? No.
Dr. Lakshmi, please wait. Wait, we're getting a mic to you.
So, okay, so you have seen the increased excitability in the postsynaptic cell.
Yes.
And just wondering why there is no changes in the astrocytes. Is it only the density has not changed, or is there any changes in the recruitment of the astrocytes? Or what about the recruitment of the NMDA receptors in the postsynaptic cell? Or is there any changes in the AMPA receptor recruitment?
Yeah,
These things would be very interesting for us to know why it is only postsynaptic.
yeah, this is an important question. That's is not due to changing the astrocytes transporters of glutamates, as we explore with the activity of these transporters. We also run experiments with the inhibitors of these glutamate transporters and astrocytes after hypoxia, and they are not altered in relation to the control group. So the changes are not due to astrocytes.
Correct.
So why only postsynaptic AMPA and NMDA receptors are affected? This is the question that we really need to address. And our major hypotheses are changes in the density of receptors, but we need to prove this. We do not have any data yet that I can use to convince you that are really changes in the density. This is our major hypothesis. As well, the characteristics of these channels because AMPA and NMDA open ionic channels, and the kinetics of these channels may be also possible one of the possible changes, but we do not have any experimental data yet that can be used for these arguments.
Okay.
Thank you.
Thank you. Any other question? Dr. Professor Malik and then Dr. Ashok Jaya.
Just compliment to our postgraduates who are here. There are two messages, He has described all the contemporary modern techniques in neuroscience, which have been utilized to find out the truth behind this. And also the illustrations are so simplified, it's very easy to understand. So I request my postgraduate to prepare their slides like this. It's exemplary, sir. Thank you very much.
Well done. Well done.
Thank you.
Professor Ashok Jaya.
Thank you, sir. Regarding the mechanism that your data that you showed, postsynaptic neurons had increased firing rates. Postsynaptic neurons in the NTS had a higher firing rates.
Yeah.
Right.
Yeah.
Now, I just want to know your time kinetics of that. How much time after sustained hypoxia does it manifest? And how long does it last?
Okay, this is a very important issue. All these experiments were performed 24 hours, so we should expose the animals to 24 hours of sustained hypoxia. We remove the animals from the chamber and prepare these lice and run the experiments. But the point is, the time window of hypoxia is critical. So the major, now we are starting to explore the initial period of time in which these changes may be even more important. And maybe in this case, astrocytes could be playing a role in the initial 5-6 hours. So 24 hours is very long time. In our view, 24 hours was not, for rats, was reasonable. But for mice, 24 hours is a long period of time. So now we need to run again this kind of experiments in the time window of 4-5 hours till 6 hours, when all the glial cells are mobilized, because this hypoxia, we must remember, bringing a serious physiological inflammatory response to the brain. And this occur mainly in the initial phase of the sustained hypoxia. So all these changes in the postsynaptic neurons, if you look, as we are planning to do this, in the initial few hours of the sustained hypoxia may be quite different.
Because the increase in AMPA and NMDA is so resembling the LTP kind of a response, long-term potentiation.
Long-term potentiation, yeah.
But the problem is LTP in the current model requires very high frequency of stimulation, about 100 hertz. And these neurons don't fire at that rate. They fire maybe at between 2 to 10 hertz. So explaining LTP by high frequency doesn't seem to be
Yeah,
the
this
answer.
is a good suggestion that we should look for the
Because
potentiation of this response with high frequency stimulation too.
because
Yeah.
your data has been used to explain the hypertension in people with obstructive sleep apnea. Because many of the OSD patients have episodes of hypoxia,
Mhm.
and then they get the asymptomatic hyperactivation. So the hypertension associated with OSD has been your rat model data is utilized to explain in humans. But now when you get a mice data, which is different, like you mentioned that there could be difference in people. Some people may respond with more hypertension, increase in sympathetic activity. With hypoxia, some may not. Given your strains of mice also have different responses.
Yeah, that's right.
So some
So
people,
this is an important point that mice, as I mentioned in the beginning of my talk, they do not present increase in sympathetic, neither increase in blood pressure. So this became even was not our purpose, became a very interesting model.
Model.
They do not develop a sympathetic hyperactivity, neither hypertension in response to hypoxia. which may be an interesting model to try to understand
Why?
why. Yeah.
Thank you, sir. Thank you very much.
Can we take some other question?
That's correct. Yeah, so exactly. So OSA patients have hypertension, and their RAT data actually was used to give the mechanism of it. Yeah. So, but then you see the mice data shows a different result. So the question is whether, yeah. So the human beings may be different responding. Some may respond with hypertension, some may not respond. That was the strange.
Any other
Thank you.
question from the youngsters coming forward? Yeah, there's one guy behind. Shival, Dr. Shival.
Sir, you mentioned current density, the calculation, sir. So it is usually calculated as current per unit area, but for the normalization, we use capacitance as the denominator. So how is the capacitance of a cell calculated, sir? I'm sorry, I'll come again, sir. Sir, for
How is? Okay, you can with this patch recording, you can evaluate the capacitance of the cells.
Yes.
Yeah, and we test that it also was not affected. So
so how do we calculate capacitance of a particular cell line that is used in the experiment, sir? Is there any standard value or it is some method of calculation?
yeah, by using some technical approaches to evaluate these changes in these cells. Yeah, it's feasible.
Okay.
Patch clamp, yeah, that's right. It's an intracellular recording.
Okay, sir. Okay.
It's a physical.
Voltage jump. Okay, sir.
Yeah, yeah,
Okay, sir. Thank you.
that's right.
Yeah, bound. So any other question from the house? So I see none. So with your permission, can I have one question, sir?
Sure.
Very interesting talk. And it's a learning moment for me. Three questions. The first questions, you started with the premise that sustained hypoxia, sustained hypoxia. Okay. Okay.
Yeah, that sounds.
So I can come to you.
It's really difficult to hear.
I can get.
Yeah, here's better.
So sustained hypoxia resulting sympathetic excitation. Some other report, it results into parasympathetic. So my question or understanding is there threshold value, like if you make changing in sustained hypoxia grades, FiO2, you keep at different levels, will the response change? If you keep changing the level of hypoxia, how the system will respond? You have used the fixed one.
No, we never try, but this is a good point, Professor Deepak. Each model of hypoxia may affect in different way all these neural circuits. One thing that I didn't mention, but is really important, that when we submit any animal, rats, mice, or even human beings to hypoxia, we have a lowering of the body temperature, reduction of the metabolism. And recently, as a physiologist, we decided to measure the temperature of mice and rats when we submit these rodents to sustained hypoxia. And mice, for my surprise, in the first few hours, they present a drop of 4 degrees Celsius in their body temperatures. A huge fall. And this may affect all the central neural control, because the neuronal activity is now, we don't know exactly how low was the temperature in the brain, but probably it was also reduced in the same direction of the body temperature. And this affects all the neurotransmission, of course. So then it's not only the level of oxygen in that we expose these animals, but we need also to look to other physiological parameters that sometimes we forget get to look to the entire system. But for example, these mice, for example, rats, if you submitted rats to intermittent hypoxia, in 10 days, they became hypertensive. I know some data from Professor Rodrigo Iturriaga from Chile that you know that they to get an increase of 10 millimeters of mercury in mice exposed to chronic intermittent hypoxia, it was required 65 days to produce a little increase in blood pressure in mice. 65 days. It's amazing. So the mice are much more, they are more adapted to response to hypoxia in a better way than rats and humans, especially if their genetic background, as I said, is from the high glands. So they cope this challenge relatively well. But you are right, you need to be very clear in all our studies about exactly the model of hypoxia that we are employing in that experiments. Otherwise, you'll be a very confusion, bring more problem than solutions to this important issue of hypoxia. Yeah.
Thank
please.
you.
Thank you.
The second question, the classical role of astrocytes, mopping out potassium or whatever metabolite. Has anybody done a negative control, like knocking out astrocyte and seeing the effect? You have tried to see the active role of astrocyte, trying to pick up potential or changes in astrocytes, but somebody has knocked them out and then saw the transmission?
No, probably not in astrocytes, because these are the first data showing that astrocytes is not, at least in the NTS, playing a major role. But in rats, we documented that astrocytes are critical for the synaptic changes in the NTS neurons of rats, but not in mice. So in terms of knocking out these astrocytes or in mice were not yet performed, at least in my knowledge. Yeah.
Sure.
Okay.
The last question is regarding the timeline. Dr. Ashok Joyal said, I was looking at the timeline, the potential produced by an MDA or this EMPA, going to 8 to 10 minutes. I was trying to calculate from your potential. So 8 to 10 minutes like an autonomic response, which means after the stimulus, it taking 5 to 10 minutes. I was wondering, is it taking too much long when you give a stimulus and then response coming up? So where's the time, like when you give a hypoxia, in how much time you get tachycardia? It's very pretty fast.
It's fast. Yes, but
How do you explain on this basis?
yeah, but look, we're talking about different moments of this challenge. Initially, are the peripheral chemoreceptors that detect this change and immediately produce some adjustments. Another thing, when you keep this stimuli, the activation of peripheral chemoreceptors for long periods of time, so then the system starts to adjust itself for the new environments. One thing is a short drop in arterial PO2. Another thing is a stable fall, and this for periods as long as minutes will make a big difference in the pattern of the response of this neuron.
Okay.
Exactly.
Thank you.
Oh,
Thank
my
you.
pleasure. Thank you. Well, here the sound was much better. I could interact easily. When you make the question from the audience, it's difficult to hear from the. Thank you. Great.
Thank you, Professor Machado, for your brilliant presentation. The lengthy discussions and interactions itself shows how much relevant it was and how much it has reached to the audience. Thank you so very much.
Thank you. Thank you.
And we have nothing to give you much except a loud clap, to the speakers, and maybe a small memento by the organizers. Over to all.
Now I respect the chairpersons to give certificate of appreciation and a memento to our resource person.
Oh, thank
Now I call now I call upon Dr. Parvati P. Patil, Professor and Head of Department of Physiology, JN Medical College, to kindly present a certificate of appreciation to our chairperson. Now we move on to our next session, that is the plenary lecture, on the topic evidence of addiction vulnerability in adolescence age due to early life stress by Dr. Professor Lakshmi T. Rao. I call upon Dr. Sameer Chate sir, Professor and Head Department of Psychiatry, Jain Medical College, and Dr. H.N. Malik, former Professor of Department of Physiology, AIMS, New Delhi, and former President of the Indian Society for Sleep Research, to kindly chair this session and introduce the speakers.
It gives me great pleasure and honor to introduce Dr. Lakshmi Rao. Dr. Lakshmi is a dear friend of us. Also, she is our General Secretary, a dynamic lady, equate with Rani Lakshmi Bai. And today she'll be delivering a talk on evidence for addiction vulnerability in adolescence age due to early life stress. She is the professor of neurophysiology and head of the department of neurophysiology in Niemans, Bangalore. And you don't need much introduction, Dr. Lakshmi. You can start your talk.
Yes. Thank you. Thanks to the chairperson and organizer. Today I'm going to discuss with you about the cause and effect, why particularly at the adolescence age, there is high level of curiosity, and sometimes it is heightened curiosity as seen, and it is more in some, and it is not well regulated in some people. So, so in order to, sorry? Okay, what should I do? Oh, okay. So we'll try to understand some basics of cause and effect of addiction vulnerability in adolescence. This is the one, I think all of you are very familiar with that. It's an HPA axis, hypothalamo pituitary adrenal axis. And when the hypothalamus is well regulated, any stimulation comes to the hypothalamus due to any of the stressors, which is one of the factor which stimulates the hypothalamus. It can trigger the activity in the hypothalamus. As a result, you see increased cortisol or corticosterone from the adrenal gland and release into the circulation. And if it is not regulated, what would happen? There is excessive release of corticosterone. It causes changes in the not only physiological functions, even the brain and behavior will be altered. So this is the topic which I'm trying to explain to you based on the evidence generated from our lab. Okay, so, and when it comes to the corticosterone or cortisol, see in animals, it's mostly the corticosterone, and in humans, we collect mostly the cortisol seen in the peripheral circulations. And this is, it is in the rhythmic pattern. So in humans, it is mostly seen in the early morning with the peak, whereas in the rodents, we see, because most of the fundamentals of this research is emerged from the animal research. So that is why I'm just showing you some example when this cortisol is or corticosterone it peaks in the animal model, which is particularly at the evening hours, it goes to the peak, unlike it is seen mostly peaking at the morning in the human subjects. Okay, when, as you can see here, there are three examples. One is in the normal SHRP period in animals, or even in the humans also. It is not well defined in humans for how many months or years it is in the stress hyperresponsive period. So what is stress hyperresponsive period? It's basically any small stresses. There is a, even though CRH is increased in the hypothalamus, there is no increase in the ACTH or the cortisol. That's called the stress hyperresponsive period. In case if there is any stresses, the first hit of the stress during the early life, particularly seen in the early life, and if there is an activation of hypothalamus, there is increase in the ACTH is seen, but not the increased cortisol. But continuous exposure to the stress, it's called chronic stress at the developmental period, it reduces the activity in the CRH, but you can see there is increased ACTH and also increased corticosterone. It means there is an overactivation of the adrenal gland leading into the increased cortisol in the peripheral circulations. So in which conditions we see this increased cortisol in the SHRP? How do we do it? We use animal model by exposing them into the maternal separation and isolation stress. Which basically we separate the pups from the mother during the entire period of the stress hyperresponsive period. You can do it for 3 days or you can do it for the entire SHRP period, that is from the P4 to P14, the postnatal 4 to 14. So what we observed, we just observed that in the animals, when you expose them into the early life stress during development period, there is no immediate increase in the cortisol is seen, but rather you see the increased cortisol at the later period, particularly it starts at the adolescence period. And interestingly, it is not just only cortisol is increased or corticosterone is increased, there is also increased dopamine is seen in the adolescence period. And this increased corticosterone, it do not stop at the adolescence period, but it continues even at the adulthood stage. Now, why it is important? So what will happen if there is increased corticosterone during the adolescence period? So this indicates that SHRP is very crucial for the developmental process. During that period, you can see that there is synaptic pruning, circuit formation, maturation, and any adverse experience during this stress hyperresponsive period, which is due to physical stress, emotional stress, or the sexual abuse, that causes the changes in the neurodevelopmental process. So when we say neurodevelopmental process, how do we measure them? So one of the best way to measure the changes in the neurodevelopmental process is looking at the milestones, developmental milestone. So, what in our lab we looked at it, one of the thing is eye opening. So because of the early life stress, during the stress hyperresponsive period, there was a delay in the eye opening. So that indicates that it's a developmental milestone has been delayed in these animals, and that could be one of the reason. Now, what are the changes are seen due to the early life stress? So there is changes in the structures and the functional development of the HPA axis, and it also alters the key regulators of HPA axis, which is amygdala, prefrontal cortex, and hippocampus. In the last several, nearly 2 decades, we try to explore this in doing different types of early life stress model. And we observed there is increased corticosterone. And we also showed that there is an impaired stress response from the HP axis by looking into activity, particularly in the freely moving animals. All these studies are done in the freely moving animals. And we saw the impaired stress response from the hypothalamus. And we saw the changes in the impaired cognition, which is due to the changes in the PFC, medial prefrontal cortex activity. And we saw the impaired fear extinction, increased anxiety and depression, reduced serotonin signaling, increased compulsivity and attention abilities, dendritic hypertrophy of the pyramidal neurons. We also saw it is not just only the prefrontal cortex, we also saw the hippocampal functioning. So like, as you can see here, we saw the impaired spatial learning and memory, increased fear generalization, increased anxiety and depression, reduced serotonin signaling, increased social novelty, not only not sociability, dendritic hypertrophy of the pyramidal neurons in the hippocampus. Similarly, we also observed similar pattern in the amygdala. Unlike hippocampus there is increased activity in the hippocampus with the increased structural, particularly in the dendritic hypertrophy of the amygdala was seen. And there are also literature talked about the functioning of the raphe nucleus was altered by increased anxiety and depression associated with reduced 5T signaling in the raphe nucleus itself. And also, we also saw there is an increased sleep period, which could be associated or correlated with the raphe nucleus functions. So these studies of previous studies of ours, which clearly showed that early life stress alters the functioning of these three regions at the young adulthood. All these changes are seen at the young adulthood until the old adult stage also. Now, the question is how it affects or impacts the adolescence age. So that is in recent last 5 years, two of my students are continuously focusing on that. Shruti and Abanti, she's here, sitting here in the audience. And they try to explore the underlying mechanisms of changes in the brain and behavior at the adolescence. And why adolescence? All of us know that that is the age we start to, we show a lot of curiosity and we take risk during that period. And we also try to, sensation seeking behavior. It's one of the predominant behavior seen at the adolescence. So this, by exposing animals into the early life stage, whether that changes the one of the behavior which we focused on, this curiosity, risky decision-taking behavior, and leading into the addictions vulnerability. And so Shruti has showed in her experiment, which is indigenously designed in our department, to test the curiosity in animals. How do we test the curiosity? So we developed the model in such a way that animals has to swim through the aversive environment, which is the water, which is filled in the water, to reach the platform where there is objects are kept, which is not rewarding. So when we tested these experiments in animals like rats, rat model, and animals which are predisposed to the early life stress, they could swim through the water very quickly, and they showed they were retrieving the objects which is kept in the unsafe zone. Now we can say that because why I'm saying unsafe zone, because they had to cross through the aversive zone. So they showed increased curiosity by retrieving more number of objects, and very quickly they were retrieving the objects. So that indicates that early life stress induced by maternal separation and isolation stress could be the reason for heightened curiosity with the increased curiosity index during the adolescence. As you can see in this result graph, so there is an increased curiosity is seen in the curiosity index at the adolescence when you are first exposed into the this chamber. Now, so till now, what we showed was early life stress. Henceforth, I'm only focusing on the adolescence age, as it is very critical for many of the developmental process. So we showed that early life stress causes increased glucocorticoids like corticosterone and increased dopamine,
and increase curiosity with the by taking risky decision-making behavior. And it could be one of the reason for increased addiction vulnerability at the adolescence age. And this could be probably due to the reduced, and we also showed that there is a reduced activity or structural plasticity in the medial prefrontal cortex, and also increased dendritic proliferations in the amygdala. And this could, changes in the structural plasticity and the behavior, whether it is could be due to the connectivity between the regions, particularly I'm going to share the data on the reward circuits. So whether there is a changes in the connectivity between the reward circuit, particularly in the limbic area. In the present talk, I'm going to talk about the prefrontal cortex and the nucleus accumbens, where there is a increased dopamine is seen. And this is just to show you that how Abanti has, this is a reward circuit pathway. She's tried to reward a task. She has done it, showing that how maternal separation stress quickly collect the reward without doing the proper assessment in the safe zone. So risk decision making is assessed based on the how much risk assessment they do it. If they do less assessment and they cross the aversive zone to collect the reward or the non-reward thing, then it is called they are the high risk takers. Anyway, I'm not going to talk to you about the neural correlates of the risky decision making behavior. Rather, I would want you to look at this data as adolescents do take the more risk, and by crossing the risky zone due to the poor or reduced assessment in the safe zone. So these animals, interestingly, they show heightened anxiety-like behavior. So this test can be easily done using several paradigms. And here in this study, we use the light dark test. So if they stay more time in the dark zone or dark compartment, it indicates that they are aversive to the brightly lit environment. They go to and stay more time in the safe zone. Of course, this is a lot of variability is there. And then based on this, so they take high risk, they show relatively high level of anxiety. And if this is true, and is there any changes in the activity patterns or connectivity between the reward circuit, and that could be one of the, is it the reason for increased addiction vulnerability at the adolescence? So this was the hypothesis. And we did the three bottle choice preference test. Particularly, we focused on the nicotine addictions. And the 3 bottles were kept, with one with the water and one with the low concentration of nicotine, then another one with the high concentration of nicotine. Then we looked into the neural correlates or oscillatory patterns in these regions, particularly prefrontal cortex, hippocampus, and the nucleus accumbens during the 4 phases of the nicotine addiction. One at the baseline, then what will happen in the first exposure to the nicotine, how this activity and connectivity patterns are altered, then chronic exposure to the nicotine from low and high concentration, which one they prefer when you give high concentration, and what are the changes which you see in that. Then finally we looked into during the withdrawal phase, how these connectivity patterns are changed. I'm not going to tell you very detail about these things, just to give you glimpses of the data which we have acquired over the last 5-6 years. And as you can see in this red one, which red bars are indicating the maternal separation stress animals very clearly, which you can see here in the slides. Early life stress has caused increased vulnerability to the nicotine addictions. So they preferred, particularly if you compare between the low concentration, N1 is low concentration and N2 is a high concentration. If you compare between them, and the moment the high concentration nicotine was kept in the bottle, and they preferred the high concentration when compared to the low concentration. So it indicates that they're highly vulnerable to the nicotine addiction, and once they're addicted chronically when you're subjected, and they prefer the high concentration, which is similar to what we see in the humans. So in this, again, the graph, which you can see here, they preferred more of the high concentration of nicotine. So this confirms that early life stress could be one of the major causative factors for increased addiction vulnerability at the adolescence. So till now, behaviorally and cognitive functions, if you look at overall, and they show high anxiety, and they also you also see that we also saw the increased basal corticosterone level and increased dopamine, and they also show the high risky decision taking behavior, and we also have some evidence to show that they prefer more sucrose when compared to the water when we give a sucrose preference test. And unlike it, so it is not the depression, rather it is they show the increased sucrose preference. And finally, it indicates that they show early life stress is one of the reason for increased nicotine addiction. Now, I'll give you just an idea about the what are the activity pattern. Particularly, we focused in this study oscillatory patterns by recording the local field potentials from these three important regions in the reward circuit, like nucleus accumbens, prefrontal cortex, and the hippocampus. So what the changes in this activity in the limbic circuit, whether that could be reason for the nicotine addiction. So what Shruti has done is she has recorded the local field potentials recording while performing the task. So this is the one, it's the local field potentials, and this is the 3-bottle choice preference test in the what's it called, addiction cage. And there are 3 bottles are kept, either low concentration, high concentration of nicotine, or the water. So, and event marker is done for every stages of the consumptions of the water or the nicotine. And this is then there are a lot of data processing has to be done before come to this level. And as you can see here, these are the local potentials. We did all the filtrations and looked into the final signals with a high signal to noise ratio. And this is the spectrogram, where you can see that theta predominance particularly in the prelimbic cortex. And of course, you also see the theta in other regions, but predominant in the control rats. So in the maternally or MS stress, early life stress animals, they show the relatively reduced theta oscillations. Of course, other oscillations are also altered following the early life stress. And this is another, the different phases. One is while crossing the risky zone. Another one is while retrieving the objects. And sorry, this is a wrong one. And we looked in the nicotine test also accordingly, marking the events while consuming the nicotine, N1, N2, and the water. So what we saw was, and there was while consuming the first exposure to the nicotine, and there is no change in the activity pattern while consuming the water. So at any frequency, either at the 0 to 4 hertz, 4 to 8 hertz, 6 to 12 hertz, and all the frequency range up to gamma frequency range we analyzed. And we did not see any changes in the theta oscillations while consuming the water. But while consuming the nicotine at the first phase, that's called the low concentration of nicotine, you can see very clearly there is increased activity at the theta frequency range in the nucleus accumbens and the hippocampus. And subsequently, that's the power spectral density analysis. And we also looked at the connectivity patterns between the hippocampus and the nucleus accumbens. As you can see here, there is a reduced connectivity, but increased power spectral density in these regions. So what does it indicate? So first consumptions of the nicotine, or first exposure to the nicotine, and there is an increased activity in the nucleus accumbens leading into it. That could be one of the reasons wherein we also saw the increased dopamine. So that could be one of the reasons there is an increased activity in the dopamine causing the beginning to show the increased preference for nicotine over the water. Then upon exposure to the nicotine continuously for maybe 20 days, first 10 days it was given only the low concentration, and the second part of that, like 11 to 20 days, we are given options for both low concentrations and the high concentration of nicotine in addition to the water. And as you saw earlier, the behavioral data, and they prefer the high concentration of nicotine. But here in this, you can see very clearly, and there is a reduced activity in the nicotine nucleus accumbens, hippocampus, and the prelimbic prefrontal cortex. So that is on the day 20 of the nicotine, wherein they were consuming more of high concentration nicotine. So the nucleus accumbens showed the reduced activity. It means, so there is a kind of, we are setting in for the like preference for the high concentration nicotine, that high is not coming yet. That could be one of the reason, because there is no activity, heightened activity is not seen in the nucleus accumbens. Whereas in the previously, the first exposure to the nicotine, you saw the increased activity in the nucleus accumbens, but upon chronic exposure to the nicotine, there is a reduced activity. And also subsequently, when you looked at the high concentration nicotine, again, nucleus accumbens showed the reduced activity at the power spectral density, particularly at the theta frequency range. And if you look at the oscillatory patterns between this nucleus accumbens and the prefrontal cortex and the nucleus accumbens, as you can see here, for both N1 and N2, there is an increased connectivity. So there is a reduced power spectral density at the theta oscillations in these regions, nucleus accumbens, but that increased the connectivity between the prefrontal cortex and the nucleus accumbens, and particularly while consuming the both at the low and high concentrations, because they're not getting the high, even though they are trying to connect to each other regions. So again, there are different types of analysis we do it to understand whether it is only the oscillatory patterns or the connectivity pattern at the waveform, or is it due to the frequency range connectivity. And everywhere it has showed that there is an increased connectivity is established between the prefrontal cortex and the nucleus accumbens while consuming the nicotine. Then what is the changes which you see in the withdrawal phase? So after 20 days of exposure to both low and high concentration, we withdraw the nicotine, and 48 hours after that, when we recorded that, very interestingly, nucleus accumbens still show reduced while consuming water, but while consuming, because during withdrawal phase, no nicotine is given, but only water is given, and there is a reduced activity is seen in that, but increased connectivity between the nucleus accumbens and the CA1 is now established, because they are going in search for the nicotine. So that is why now hippocampus got involved, not the prefrontal cortex. So that could be one of the reason for increased connectivity between the nucleus accumbens and CA1 is seen, but not with the prefrontal cortex. So far, we showed that there is, during the first exposure to nicotine, we specifically saw the increased power spectral density at theta frequency range in nucleus accumbens CA1, but not the PL, the prefrontal cortex, while consuming the low concentrations. And it is associated with the reduced synchronization between the nucleus accumbens and the hippocampus. Then after chronic exposure to nicotine, there is a further reduction in the power spectral density in all the three regions. And while consuming the high concentration of nicotine, we saw the reduced power spectral density in the nucleus accumbens, and with an increased synchronized oscillations between the prelimbic cortex and the nucleus accumbens. And of course, there are many analyses we have done, granted causality. Of course, that didn't give a very clarity. And upon withdrawal, there is a reduction in the activity in the nucleus accumbens continued, but increased connectivity between the nucleus accumbens and the CA1 has increased. Now, so these connectivity patterns, how that has affected the dopamine level. Yes, I'm just 2 minutes.
Just 5 minutes.
I'll finish in 2 minutes. So there is no changes were seen in the D1 receptors, and DA concentration, dopamine concentration is reduced, which is comparable to the reduced activity in the nucleus accumbens with an increased D2 cells, D2 receptors in the nucleus accumbens was seen in both the hemispheres. So with this, I conclude that curiosity is one of the hallmark of behavioral phenotypes of adolescence, and early adverse experience could enhance the curiosity like behavior at adolescence, enhance the risk for substance abuse disorders in adolescence age. And this could be possibly due to the changes in the dynamic interactions between the limbic circuit, and that could be responsible for nicotine addictions at the adolescence. So dampened HPA axis responses during the SHRP, particular stress hyper-responsive period in the animals, we define it as a P214, and the humans mostly it is defined as a 0 to soon after birth until 3 years of old. And that is responsible for the well-regulated risk assessment, increasing the well-controlled curiosity and the risky decision-making behavior. Maternal separation stress could alter the dendritic morphology of the limbic regions, and associated with the increased curiosity and the risky decision making behavior, leading to nicotine addiction. And upon nicotine withdrawal, there is a reduced dopamine, which could increase the sensitivity to stimuli, contributing to the negative feelings and the relapse. So the work is carried out in my lab in the last 5-6 years. And the major contributors for this research and by the Abanti and Shruti. Shruti has done focus mostly on the nicotine addiction and the curiosity. And Abanti has also looked into the curiosity by looking at the risky decision making for the rewards. And of course, this is my current lab members. There are so many. So in this, I would like to say that prevention is better than cure. So it's better to take care during the early life for the future, bright future in the life. So thank you. So in order to have a very nice life, I think we need a smiley faces at the childhood level. Thank you.
Thank you, ma'am. Very well presented. Very impeccable presentation, I would say. You started with basics, including the HPA axis, then you went on with evidences, then its application clinically. Everything is covered in this. And more importantly, you maintained the time. That time management was wonderful. And we have 2 minutes more. We can open this floor for discussion. We can invite some questions, couple of questions from the audience, please. Yeah, there we have one. Somebody give him the mic.
I can hear you, then I can. This is an animal behavior. So normally P30 to P50 or up to P60, we can postnatal, I'm saying. Postnatal. Yeah, postnatal, 30 to 60, we consider it as an adolescence age.
If we are doing adolescence research, we have to take permission from parents. Otherwise, it's a vulnerable age group. So then.
And we have an ethics committee. So we do this experimental procedures after obtaining all ethical.
So in this, we not required to take permission from parents of that animal.
Yeah,
These are vulnerable age groups, so for the clearance of
that's a
study.
good question. Maybe I want to talk to the mother. Yeah.
Second question was, madam, generally the seeking of the risky behavior or that addiction seen in in a human being, there is mostly though the depression patient used to get kicked while taking the drugs like that thing. So have you, in initial phase of your animal, they're normal animal or a depressed animal, have you rule out the depressed animal? Otherwise, in depressed person, already there is a seeking from the addiction is there.
Correct. So that's a good thing, good question actually. But very interestingly, maternally our model is like, maternal separation and isolation stress. And these animals, very interestingly, they did not show the depressive symptoms. Rather, they showed the sensations, like all our previous experiments when we had done it, classical features which we tell for depression, it was not seen in these animals. Rather, they showed increased spatial cognition, they showed increased fear cognition, they showed, of course, there is an increased anxiety. But as again, depression is not necessarily always associated with the anxiety. There is a dual response seen, but lot of features which showed the opposite of the depression. It is not the depressive symptoms. Yeah,
Thank you. Dr. Manish Parwar, SNMC Juba.
thank you.
We can have one more question from the audience.
Okay, if there are no question, probably I'll.
Yeah, I can't see there. Is anybody that side?
Thank you, ma'am. Just wanted to ask that culturally we see a lot of difference in this. You said 0 to 3 years at a maternal separation period. So culturally we see that some cultures are having that kind of practice while other cultures follow no maternal separation during that early age. So is there any evidence available culturally in human beings regarding this thing, the addiction in later life in different cultures?
Culture wise, maybe I'm not able to answer that question because we didn't explore that. Anyway, this everything is mechanisms based. We do it in human studies.
So I think sir was asking about humans. So I think to explore it further, we can see
Yes.
how different cultures are having this addiction rate.
I only wanted to find an answer for that. Why each one is different? Why some are more vulnerable? That's the one. And I wanted to find an answer for that. What is the underlying mechanism? Why some are showing more vulnerable? That's the only reason. That is why we used early life stress as a model. So it is
Actually,
only the
my
mechanism based. Yeah,
My
not.
question came from your last slide, that happy smiling children. But
Ah.
I think it is very difficult to project it to humans because of so many other factors which
Yeah.
come in over life.
I would like to
Okay. Yeah.
contribute to
Yes.
this.
Yes. Yeah.
Yes, you are right, ma'am. The behaviors do differ. Basically, the behavioral disturbances or the changes in psychiatry, they're culture specific. So change in culture, there is change in addiction behavior also.
I think if we regulate the HPA axis, well regulated HPA axis at that critical page, probably at the adolescence, again, if you regulate that activity during multiple parameters, it's not only parenting, even the school curriculum everywhere, give excitation and other positive things. That would take care of the addictions for something. Let us inculcate the addiction for learning something which benefits the brain and behavior. So that is the reason I said prevention is better than cure. So it's our responsibility to control this HP axis functions at both period. We know the rewiring takes place particularly at the adolescence. We have the second chance. If we miss the boat in the first 3 years, probably we come back at the adolescence age, have a well-controlled, not that punishment or anything, well-controlled, well-regulated HP axis function. With that, maybe we can direct the prefrontal cortex to that right direction.
Thank you so much.
Dr. Lakshmi,
Yes.
I have one.
Yes, sir.
Uh, In the present society, we have the mothers, most of the mothers are working
Okay.
and the kids are at home.
Yes.
So what kind of message you want to give to the society?
Yeah, so that is one of the challenges. Of course, we all go through that as a mother. But this can be taken care very well by well-planned activities like engaging the children at the adolescence age, mostly with the physical activity. So school current curriculum is focusing mostly on the, getting into particular course. So as a result, we'll focus more on the academic one, not on the non-academic activities. So if the mothers engage their children, that's why we got a two chance at the early life, and the second phase is at the adolescence age. Adolescence age is, you can direct them because they seek for the sensations. At that time, engage them in the more of physical activity, which I always I emphasize more on the physical activity. That gives a similar addictions. It is not any of the substance addiction, rather they get addicted to the play and get the reward. They want the reward. So first 3 years of life, of course, now maternity leave is given for 6 months. I think that should be enough initially, but afterwards, of course, the house environment should be kept very well regulated. There is no question on that. Maybe good daycares would take care of it. Yeah.
I think
Thank you
there are
very much. Yeah.
no more questions.
Yes.
Thanks for a wonderful presentation, and you deserve a big round of an applause. Thank you, ma'am.
you.
And I thank the organizers for giving me an opportunity to chair this session. Thanks a lot.
I request Chairperson to present a certificate of appreciation to Dr. Thank you, sir. Now I request Dr. R.N. Raichu, Professor and Head, Department of Physiology, USM, KLE, International Medical Program, to present a certificate of appreciation to the chairpersons of this session. Thank you, sir.
Now give us a couple of minutes as we set up for the next session, that is panel discussion. So as we are lagging behind, we will not break for tea now. After the panel discussion, we will have tea break.
Next, we move on to the panel discussion on the topic From Blackboard to Broadband, Teaching Physiology Across Generation by Dr. Richa Gupta, Professor, Mahatma Gandhi Medical College and Research Institute, Pondicherry, and Dr. Utpal Kumar Datta, Senior Consultant, Professor of Physiology, Manipal Hospital, Kolkata. Dr. Anita Herur, Professor of Physiology, S. Nijalingappa Medical College, Bagalkot. Dr. Vital Kode, Professor, SDM Medical College, Science and Hospital, Dharwad. Dr. R. N. Raichur, Professor and Head, Department of Physiology, USM, KLE. Dr. Dipali Kadek Angadi, Coordinator of NMC Nodal Center, Professor of Community Medicine, Jain Medical College, Belagavi.
Teaching physiology across generations. So I would like to introduce our panelists. Firstly, all of them have a vast experience of teaching physiology across generations. So we have with us Dr. Utpal Kumar Datta, who is a senior professor and has experience of teaching from generation X to Z, including generation Alpha. So sir is working as a senior consultant and a professor of physiology at Manipal in Calcutta. And then we have our beloved Dr. R.N. Raichur sir, who is so enthusiastic personality and also the professor and head of USM KLE International Medical Program. So sir is very active in medical education. And then we have my friend, Dr. Anita Herur, who is a professor at SNMC Bagalkot. I admire her myself because of her commitment and intellectual capacity. And she has published so many innovative teaching learning methods in medical education. Thank you. And then we have Dr. Vittal Khode. So I'm proud to say that he's my undergraduate batchmate and also a professor presently working in SDM Medical College. And he has published books on case-based scenarios, especially the case-based scenarios on MCQs. And sir also has his own YouTube channel. Again, another person we have, Dr. Richa Gupta, who is a very vibrant online. So madam has her own YouTube channel known as Open Physiology. And then we have her own app also. So madam has published books in practical physiology, especially emphasizing on OSPI and OSCE in physiology assessment. Okay, so I just welcome all of you for this panel discussion, and then we'll shortly start with the discussion. Thank you. So the topic is from blackboard to broadband, teaching across generations from the baby boomers to generation X to Z, millennials and generation Alpha. So for today's generation, so I think Dr. Utpal sir can highlight on his teaching experience as he's involved with teaching across generations on blackboard teaching. Sir.
I have been in teaching since 1980, since last 45 years. And I have seen the evolution of the different teaching aids, starting from Blackboard to broadband. And mostly during our student life, as well as in the major part of my teaching career, we didn't know anything except the Blackboard. So we used to teach comfortably with Blackboard. We never thought of that we have any problem in teaching with the Blackboard. Sometimes there is problem that more material is required. In that case, we have just prepared another extra Blackboard, drawn something on that Blackboard and carried that to the lecture class with help of our students like that. I never felt any difficulty while I was using the Blackboard since so many years. But in recent times, for last about 10 years, I've gradually switched over to Blackboard and now only Smartboard. Now you see, so I have seen everything. There are many advantages of the Blackboard, no doubt. But once I have started using the other audiovisual aids, I have just learned what were the disadvantages of the Blackboard. Blackboard was advantageous, it's very simple, very easy to use, and very cheap. Electricity is not required. But there are problems also. Lot of disadvantages are there. See, first is your just eye contact is lost when you move towards the Blackboard. Then students are left uncared like that. And your space is limited. You can't write more. Your handwriting may not be legible. Students may not be able to see. And these days you see in a medical college where 200, 250 students are being admitted and students sitting in the distance, how will they see the Blackboard clearly? Your handwriting, and that handwriting must be legible. I don't know, the handwriting of many of our teachers may not be that legible, and it doesn't become legible. Why? Because see, you have to teach in a limited time, and then you have to write quickly. And if you write quickly, it can't be, it really becomes very difficult for the students to note. So all these things were there, but I never complained of like anything. We continued like that. But once we started using the PPTs and other mode of other audiovisual aids, which I've started during the Corona period, suppose. If our teaching was entirely online and we had to do the PPTs and we had to do a lot of innovations how to teach the students. So we have managed to do, and now we can understand what are the advantages of all these recent audiovisual aids. And really these are very good. And there may be some disadvantages, but all are very advantageous, no doubt about it. And recently in the college where I have been teaching, there is no Blackboard, only the Smartboard is there. And sometimes I feel difficulty. Why? Because when I want to illustrate something, there is no Blackboard or whiteboard even. So it must be there. It must be present in the lecture class or any class. So it should be blended with other audiovisual aids.
Okay, thank you, sir. Sir, as we know now that animal experiments are banned for demonstration and teaching. So what is your opinion on relevance of using animal experience in teaching physiology for undergraduates and postgraduates?
See, for undergraduates, these days particularly, animal experiments may not be done because time is very less. They get maximum 7-8 months. Out of 1 year, lot of time is wasted behind counseling, that examination. So 9 months maximum they get. Within that short span of time, it's difficult to do the experiments with animals. Rather, computer-assisted learning, which we do with the department in collaboration with the Department of Pharmacology, that is enough. But for the postgraduates, animal experiments must be there. When we were postgraduate students, we did a lot of experiments with rat, dogs, cats, etc. And see, to learn the actual physiological phenomenon, animal experiments must be done by the postgraduate students. And not only that, see, postgraduate students have to learn how to do research. And in the last two lectures, what we have noticed, all those research work was based on the animal experimentation only. So when will they learn the animal experiments if they don't do in the postgraduate part? So animal experimentations must be given. The postgraduate students must do the animal experiments. And the ethical issues must be tackled care of like that. What the last time somebody just pointed out that point. No doubt there are some ethical issues. Considering the ethical issues, the animal experiments have to be done by the postgraduates. This is a must.
Thank you, sir. So now we'll move on to Dr. Raichur, sir. So please share your experience on PowerPoint presentations.
Thank you, madam. The physiology teaching has undergone a tremendous transformation over multiple generation of the students from Gen X to Gen Z because of advancement in the technology. So over the years, the blackboard teaching was shifted to OHPs, then to the PPTs. The PPTs have become now very popular because compiling and running a presentation becomes very easy. But there are some skills and techniques which are involved in an effective PowerPoint presentation. These skills and techniques I learned from Dome by various workshops and also from computer experts and also from the experiences which I have gained through my previous classes. The techniques which I learned include how to incorporate our creativity in the presentation. Second is, we have to design the slide after careful planning, condensing the information into keywords and using our narration to expand the concepts. Then we have to design the layout of the slide for readability and aesthetic purpose by following rule of 6, using sans serif fonts and the font size greater than 24 and contrast, text and background colors and proper placement of graphics and photos. And most important is creating animations, transitions and incorporation of external video into our presentation. So the first slide should contain the learning outcomes or objectives, and the last few slides should contain summary, the references, and the references for further reading. The presentation skills which are learnt include maintaining eye contact and using progressive disclosure and pointer and not obstructing the projected image while walking on the dais and creating a transition to bring back the attention of the student if the lecture is long. And most important is rehearsing, practicing, previewing and timing the presentation. So with this, I was able to prepare and present well and continue to improve every year thereafter. Thank you.
Thank you, sir. Sir is working in an international medical program, school. So sir is having an experience of teaching in problem-based learning. So we want sir to share his experience on problem-based learning.
Thank you, madam. The physiology teaching has undergone a profound change over the decades because of evolution in philosophy of teaching, innovations in pedagogy, and changing needs and learning preferences of the students. Hence, there was introduction of an active learning method like PBL. The PBL is better than the didactic lecture because it is integrated, interactive, and student-centered learning. Although I have undergone training for PBL, irrespective of it, PBL was really challenging task initially because I was accustomed to didactic lecture where the teacher is the central authority who is delivering the lecture and assessing the knowledge. But in PBL, we become facilitators who are going to guide the discussion rather than directly providing the answers. So I have to develop patience and the skill to listen actively rather than dominating the discussions. And I have to develop the skills in group dynamics, communication, most important, conflict resolution during the discussion, and time management, etc. Irrespective of all this problem, I was really satisfied when I saw the students in the PBL taking the responsibility of their own learning and integrating the basic science knowledge with the clinical scenario. And the PBL helped in my personal growth also by improving my clinical knowledge and reasoning abilities. PBL keeps me updated with the latest medical knowledge because in PBL, the students bring diverse references and resources. So PBL helps in lifelong learning attitude both in teacher and the students. And I experienced a closer relationship with the students in the PBL. However, I faced many challenges also because the PBL is more time consuming than traditional lectures in preparation. And it is very tricky to modulate unequal students' participation during the discussion in PBL. And it is very difficult to suppress the urge to give correct answers during the discussion in PBL. And there are problems in assessment also because the PBL is assessed based on many components like teamwork, then reasoning abilities, lifelong learning, self-directed learning, etc., which is more complex than the Viva examination. But with all this, I feel now I have mastered the art of PBL facilitation at least partly. Thank you, ma'am.
Thank you, sir, for sharing a rich and practical experience of problem-based learning. Now we move on from the traditional methods to as the NMC is recommending us now that 2/3 of the teaching session should be the small group teaching. So I request Dr. Anita ma'am to highlight on innovative teaching methods which we can use in physiology for teaching undergraduates and postgraduates.
Thank you, Dipti ma'am, for that question. I belong to Gen X, and it was comfortable to teach Gen Y with the PPTs and blackboards as Raju sir and Utpal sir highlighted. But then the problem was when we started teaching Gen Z, there was less amount of enthusiasm and more availability of the internet facilities and the more online resources that made them sit less in the class, I suppose, less attentive in the class, I suppose. So it was a challenge for the teacher to resort to something else. So then that we thought was because also the syllabus was changing or the curriculum was changing from traditional to CBME, and also our approach would change from a teacher-centered approach to a student-centered approach. So there we thought of blended learning. We had to resort, there was no other option. Blended learning was the one we would resort to. So in which we taught a class, regular class with interactions of the basic concepts. And then we also switched on to something which was very inquisitive for the student, like giving them the online quizzes, okay, and also assignments. For example, I would take a class on pulmonary function test and give them an assignment to interpret pulmonary function test and then post them in WhatsApp groups, or it could be our Google Classrooms, which were free then. But then now we resort to our own LMS for assignment submissions, etc. So that was a bit active learning that we engage the students in. Then we also tried flipped classrooms, wherein we gave the basic content, like for example, the normal cardiac output. So we gave our own PPTs also we posted in the WhatsApp group. And then when they come to the class prepared, read about it, then we would discuss more complex things like why the ejection fraction in this particular case of CCF is reduced, and how are the drugs effective in recovering of this patient, how it would increase the ejection fractions, and also the operation of Frank-Starling's law, for example. So many of the complex things we would discuss, giving them the basic knowledge at their own pace. So this was our experience in flipped classroom. And also we incorporated at PG level some blended learning, like in journal club presentations. Apart from the normal journal club presentations, we asked the PGs to interact online with the authors. The authors were well reputed with having published their own work in reputed journals. So PGs had interactions online so that they would discuss their methodology, especially the research methodology, techniques, their limitations, as well as advantages. So, of course, it was written in the journal and they would critique it. But then this motivated the postgraduates to take up research, and also they would be happy, and they have their own place in amidst the researchers, other researchers in their own field. So that would really help and motivate the postgraduate students with this blended learning. And also we incorporated a number of active learning techniques like claim modeling to teach them CNS tracts. And also we took help of the senior peers in reciprocal peer-assisted learning. So there are many number of active learning techniques or strategies that we can implement. And many of the teachers have already done, I know, but then it is left to the creativity of the teacher so that we make the student more engaged and keep him there for the purpose of learning. This is my experience, Dr. Dipti.
Thank you, madam. So I just want to know your opinion on use of virtual reality and augmented reality in teaching physiology.
Yes, ma'am. This is the need of the hour, the AI, the AR, the VR. And this is the need of Gen Z or Gen Alpha and Beta in the near future also. So it's like the students are going to AI. Yes, we are there. The students are going to social media. Yes, we are there. We are going to virtual reality and game zone. Yes, we are there. So it's just like your Vodafone and the Park Dog advertisement. Wherever you go, our network follows. Okay. So wherever the student goes, the network of teachers have to go. It has to go. Okay. Then only we can make the learning more impactful, I suppose. So talking about augmented reality, yes, there are a number of animations, videos which tell us about the physiological processes. I agree. And we can incorporate them in our day-to-day activities, teaching, learning activities. But then virtual reality, I don't find enough of them to be used for the learning teaching purpose because the authenticity lacks. So even students are more attracted towards gamification. So we can combine gamification with virtual reality, but then the authenticity is again a question mark. So we need to work on this with collaborations with our AI engineers or computer engineers so that the authenticity remains and the physiological processes are well explained, because what they experience as a virtual reality, they will never forget throughout their lifetime. So when we are inculcating in them these things, physiological processes, the onus is on us to keep them more the information more authentic. So that's my experience about these things. Thank you, ma'am.
Thank you, ma'am. So we'll move on to Dr. Khode. Sir is having a vast experience of running his own YouTube channel. So we want sir to discuss about the different online platforms. The students are using different social platforms. So what do you recommend for students, and what is your experience?
Thank you, Dr. Dipti. Now we all know that importance of blackboard teaching, OHP, PowerPoint presentation, flipped classes, problem-based learning in a setting of classroom. These methods are well structured and students can engage with material and interact with their teachers and peers. We have to remember that this generation belongs to the Generation Z, and this generation has exposure to the internet and mobile devices, which has led to significant increase in online learning and making it as an integral part of the modern education. So we can say that digital learning has become a boon for this generation. We have so many digital platforms, including social medias like Instagram, WhatsApp, Telegram, Facebook, Twitter, and YouTube being most important one, which offer their educational contents almost for free. In all social medias, we can see these structured video lectures with animation where you can view them, review them at any time and at any place and make sure that your concepts are clear. And you can learn at your own pace. And you can search the topic of your interest with the keywords or the playlist, and they are available instantly. Shorts are very useful and they can make you understand the topics in less time, and you can revise it very fast, which are very important during examinations. Flashcards and integrated diagrams are game changers where you can memorize and understand the complex physiological processes. YouTubers also offer crash courses where they can teach the systems like respiratory system, blood in 2 to 3 hours, and it is a game changer for the students. Most of the YouTubers also, they solve question papers, and it can aid to your exam preparation. And you can also solve MCQs online, and instantly you know whether your answers are right or wrong, where you can assess yourself, where you are lagging behind, and where is the scope for your own improvement. I have also learned a lot from teaching online on YouTube. This has been a very valuable experience for me. By observing different YouTubers and their teaching methods, I have improved my learning and teaching skills. What I understood is that YouTube helps immensely teaching the fundamentals of physiology, where it can be very useful for the students for learning complex processes of physiology. Another important thing I learned is how to interact with the students. It is not only important to give them answers, but it is also important to give them a constructed feedback. Even sometimes I have adopted my teaching methods based on my students' feedback. Another thing, what I have learned is creativity, especially in drawing suitable diagrams, which are simpler and understanding to the students. I have learned to be patient and empathetic towards students, which are essential qualities of effective learning and teaching. Yes, in my opinion, the YouTube learning can be game changer if it is adopted effectively. Thank you.
Thank you, sir. So now moving on from YouTube to, we have Dr. Richa Gupta, who has a first-hand experience in building the learning management system. So I request Madam to share her experience on this.
Thank you, ma'am. Learning management system is a hosting software where we can share the content with the students. If we see YouTube is also a hosting place where unofficially many teachers are sharing the content. Now, the problem these days is that teachers tell students not to watch YouTube. So I have seen many people sharing that, and when I'm sitting in that forum, and I am also a YouTuber, I have a YouTube channel, Physiology Open, and I tried to make it official as an app, which also is in the name of Physiology Open. Paradoxically, I started this YouTube, that is the unofficial version, when I was working on official version of my Institute of Learning Management Systems, because we faced a lot of challenges for sharing this content in learning management systems. When you want to make the everything organized, you are looking into the curriculum, you are looking into the learning objectives, you are looking into the assessment, and everything should be matched in the learning management system. That is how organizations deliver the curriculum to the students. But because of the obstacles, I moved on to YouTube because the content creation itself was the first problem which any faculty faces. So I thought, let me jump into the river myself and see what are the where are the obstacles and how to face them when reaching out to other faculty to make the content. So that's how my YouTube journey started. Coming back to learning management system, that where we succeeded and where we failed. Actually, one of the most important thing in learning management systems, which I foresaw, was that students can access the content anytime, anywhere, at their own pace, in their own learning style. So that YouTube and other platforms, which Sir has just now shared, there were you can share different forms of the content. Some place you can share videos, some place you can share audios, but learning management system offers a place within the organized forum where you can share the contents in all different types of format. Let it be videos, let it be podcasts, let it be text, visual, even the interaction. You can increase the interactivity. So suppose when we are creating the videos and you are watching, student is watching YouTube videos, or even the videos which are uploaded in the learning management system, there is one very important danger of passive learning. We don't know how much a student has learned. So these days we have the technology of interactivity where we can insert the MCQs or questions, even short answer questions within the YouTube, within the videos, and only when the student answers the question, then he proceeds in the video. So the teacher will get an instant feedback that how much the student is actually going through the content. Because one of the main problem which we faced is that teachers were asking that why we should do it, student will not even watch it. So we get a straight feedback that how much time student has spent on the LMS platform, how much content he has accessed, how much questions he has answered. And once the content is created, the teacher need not correct the assessment, evaluate the assessment manually, because even now these days, even the short answer questions can be evaluated automatically. Looking at the keywords. So MCQs, we know that yes, everywhere softwares are there and that can be evaluated. But even the short answer questions, like 3-4 line question, like very common is physiological basis of understanding of certain things, that can be asked and automatic evaluation can be done. Yeah, somewhat we have to keep an eye, because blindly we cannot trust the technology like that. It is not at that stage right now, but yes, it gives a lot of opportunities for teacher to track students progress. So that is one thing what we thought that yes, LMS should be done.
Okay, thank you, madam. So what is your suggestion to the best practices while building an LMS? So what are the best practices institute should follow? And also how you have addressed the faculty challenges? You have shared some of the experiences. Any other experiences of sharing how to overcome the faculty challenges?
Yeah, coming to the best practices, one thing is that when we are creating the content, we are focusing, as teachers, we are focusing only on the physiology part, but we have to look at the technical aspects as well. So in the 5G generation, currently we are in 5G generation, but reality is that even in medical colleges, we don't have, in private medical colleges also, we don't have that much connectivity. And we have to think of the farthest student who is staying in the periphery also should be able to access. If we are providing something, then it should be uniform access to everybody. So that thing we have to take care. If the college is providing, they should provide adequate internet bandwidth for access of the content. The LMS should be mobile friendly, because in West every student uses laptop, but in India students are using and accessing content on mobiles more rather than on iPad or on mobile on laptop. So the content should be mobile friendly. The file size which is uploaded, that also matters. If we are high definition videos, ultra high definition videos we record, and they don't play in the internet bandwidth which is available, then it is of no use to the student. So we have to ensure that the file size is adequate to play in the internet connectivity which we have. Then obviously we should not make it purely passive learning. We try to add interactivity at whatever level we can. And then training for the students and teachers is required for LMS handling. because we know that all teachers technically we cannot be at the same level. The world is running very fast. Each day new app, new something is coming, and if we start running after that, then we will go nowhere. So we have to clear the concepts that where we will stand, what we will deliver to the student, and we should give uniform training to the teachers. And for the students as well, we should give the training, because even though we hear a lot that they are Gen Z, but the reality is in when we trained a student, I personally trained a student, we found that they didn't know where to look at the screen and where to hit that click button also. They just want instant answers. So we have to really be hands on in training them. So that is another thing. Coming on to faculty challenges, yes, content creation is a huge challenge. We can expecting everybody to create videos, it is very difficult. Nobody, it is like almost next to impossible. So yes, teachers should do best what they are good at, that is focus on content. Delivery, they can try like the lectures, but we should get organization support in technical aspect, in recording, in editing, and wherever needed, the training if the teacher needs. So that is very important. With coming of AI, it has become very easy, but with the use of AI also, we have to be careful that we are using a student friendly language. If we see the AIs of today, they are using mostly West language. So we have to train the AI to use a student friendly language to make it more accessible to the students also. And we have to keep an eye on the content which is generated by the AI. And one thing extra I want to add, because NMC these days is stressing on competency based education. So one of the point which comes is that how to map this competency and how to assess this competency. So in this case, both for undergraduates and postgraduates, it is a boon. LMS is a boon because it will technically map the performance of the students over time. You can map the learning objectives with the content which you have created and with the assessment, and then see that how the students are performing, and it can give you a map of the competency of the students in various domains. In fact, in our college we are using it for the postgraduates, where we have created interestable professional activities, competencies in various domains. We have created milestones for each domains, and we are mapping actually that at what stage of teaching they are at which level. And that is possible because of learning management systems, where we are giving constant feedbacks. That is mainly for competency, we are using feedbacks, and we are compiling the responses and getting the competency score for the postgraduates as well. So that is from my side, ma'am, on learning management
I
system.
think, madam, even multilinguistic and
Yeah, one thing more that these days NMC is stressing on multilingual delivery of the content. So again, in lectures, it is not possible because of the variety of the students which we are there. But we can add that kind of content to the learning management system. We can even curate the content which is available on the YouTube in different languages. So we, in different colleges, we have faculty with different language ability. So they can be chosen and they can choose particular content from the YouTube also. They can take out clips also and give it to the students, which can be given on learning management system. So these are some what NMC is recommending. LMS is a boon to implement all that things.
thank you, madam. So I invite any questions from the audience for our panelists.
First of all, congratulations for your own LMS, and we will like to learn it more after the session is over. But regarding internet, this is a mindset of faculty as well as student कि कॉलेज दे रहा है तो कॉलेज को ही नेट देना है, खुद के लिए व्हाट्सअप यूज कर रहे हैं, ईमेल यूज कर रहे हैं, तो उसके लिए खुद का नेट यूज कर रहे हैं. तो एलएमएस और यूट्यूब के लिए भी फैकल्टी स्टूडेंट बोथ कैन यूज देयर ओन इंटरनेट, क्योंकि सब कुछ अगर हम इंस्टीट्यूट पे डालेंगे तो गवर्नमेंट में और कई कई जगह प्राइवेट में भी वो डिफिकल्ट रहेगा. और जैसे सर ने बोला है कि दिस इज जस्ट माय सजेशन कि एसडीएल तो बोला ही है एनएमसी ने. सो व्हेन स्टूडेंट आर हैविंग देयर ओन thought wise YouTube, rather we suggest they go to this YouTube and look at these YouTube channels. Thank you.
That's what I also say. Don't tell the students not to watch, tell them what to watch.
AI and technologies here was there and going to stay here. So we have to embrace it. Thank you.
Thank you very much. Lovely panel. I'm happy to hear all of you, madam, when you're talking about. So I have two quick points. One, I think I endorse what you said, what to watch and what not to watch. But two quick points, like, uh, well, it's a debate, but do you really think only the internet version or only the classroom version? I think that we have to be talking something in between, because the classroom version, uh, I, being the physiology teacher, would always feel that there's a different connect. However, as you all have spoken, however, the internet has got its own ups and downs. To you, what do you say? Yeah. Would you like to keep it 50-50? Would you like to keep a little more up and down depending on the technology input? Because if AI is a powerful tool, Claude would do everything for us, but we are not depending on Claude, right? Over to you.
Yeah, thank you, ma'am, for the wonderful question. I think I missed that point. Actually, internet learning management system, they all provide a great way of blended learning. They provide a way for teachers to do much more than they ever thought. Because in lectures, as it is known that lectures are for average students, lectures you cannot target in depth entire thing, and for that NMC suggests small group teaching and all. So why not include these recorded lectures and LMS to let students have their own learning? You are giving them their own pace also. So same 20 minutes lecture they can watch in 40 minutes, and in class use in class time more for in-depth learning, like what we call flipped classroom. So that is also a technology which will give us a pathway for implementing flipped class.
Appreciate it. Actually, in person, eye to eye contact and solving very trivial points, that still remains a strong power to it. And what somebody was telling that about the internet and what exactly is a take of students, if anybody can just tell me, in the medical colleges for physiology? I know about the veterinary physiology students and animal biotechnology students. What's the take of medical physiology students? They are bent to class, or they're bent to the internet? Maybe you may not answer the question if you don't like, but I was just curious to understand. Thank you so much.
It's a valid question actually. I think anybody gets.
Current students, that students are using it, and when you at the end of exam, final exam, uh, they answer in such a manner sometime to a question that you ask, सही में बताना कहाँ से पढ़ते हो? मैं कुछ नहीं करूँगा, मतलब मार्क के बारे में मत सोचना. So the most of students have answered. I have specifically asked this question because I wanted to know this, that हम सोचते अलग है but practical कुछ अलग है, होता है ऐसे. So that way, what answer I got was, they learn at their pace, as you said, just before exams, or very few people are really behind the understanding of concept because of their pressure for preparing for NEET PG. So current MBBS degree and current physiology passing, their agenda is different than what we think of understanding of subject in depth. That may not be there. So our role as a teacher, I feel, motivate them by demonstrating or behaving in that way, that by you have to understand the concept that will help you in your NEET PG also. So that is a challenge, because whatever you say, SDL karte hai, small group karte hai, PBL karte hai, OSCE karte hai, but are they into it? Really, are they into it? And do they want it? So that is another question. Thank you.
Okay, I think this would be the last question. Sir is waiting from a long time.
I'll just, can I answer sir's
Yeah.
question? I agree and disagree with sir little bit, because from my experience, I teach only concept on YouTube. My all videos are concept based. YouTube, and I get a great feedback from students that they are doing in-depth learning. And before exams also, my YouTube channel peaks, the views peaks like anything. And they text me personally also that the concept which they have not understood for years, even final years are watching, even MDs are watching. They are telling that they were struggling to understand this concept for years, and they had taken an escapist attitude with that. Now they have understood. So if they are given a channel for that, I think they may use it. We have to trust also little bit. That's my point of view. It may vary.
So, this would be the last question.
So first of all, thank you very much for, because personally I use blackboards to YouTubes. And I believe that you have to use all that is available for the best of the student. Upfront. The query which I was looking now, the generation is, I'm telling you, the way YouTube was there, today it will be AI. It will overrun everything. I am personally using AI and it's amazing for even deep research level. So how do you foresee? Because the way YouTubes have changed the teaching, the way books changed teaching. So teachers, books, internet, YouTube, now AI. So what's your take on that? Just your views on that.
Thank you for that next very nice question, sir. I was very much, I wanted to answer the same thing, but the question was raised here. As a part of medical education and a medical educationist, I should say the teacher is 80% and 20% is the rest of the things, whatever aids we are using, whether it is Blackboard to broadband, anything for that matter. I would give an example, my pranams to Dr. Putturaya sir here, on whose name Best Teacher Award is given. He was my teacher. Sir will not remember me. I remember sir 35 years back. But then what sir is an excellent teacher, all of us know. And still I remember the concepts of physiology and I remember his actions too. What actions did he do when he was taking physiology, for example, on the urinary bladder? I still remember, sir. And the actions, the humor that he has in his class, the touch of humor, I still remember that. And I will remember it for lifetime. And that is what is in medical education. 80% is you and the 20% is the rest of the aids that we are using it. And it is you that who will decide what to use and what not to use, and how a student will be incorporated into that active learning process and how he is going to be benefited. And for all these process, I think the small group discussions will be of more help to understand the mentality of the students as to which student does need what and to what level. So it is we who are going to decide it. In material of what VR is coming or gamification is coming or simple Blackboard also we need nowadays. That's what I feel. Sir. Thank you.
Now, just I like to add one point. Just I'd like to remind all of you that as per NMC regulation, the attendance of the students must be 75% in theory classes and practical 80%. This is the NMC rule. So students must come to the college and students are to attend the classes, and the classes are there for the interaction with the teacher and students, and the students are, and these days we have to follow the learner oriented method. For that thing, we have to blend all the probable audiovisual aids starting from Blackboard to broadband. This I want to say. Thank you, ma'am.
Sorry, I will just add a little. Actually, we have to use a blended learning for these people. The teaching in the classroom is definitely rewarding, but the self-centered study, self-directed study is also going to be important. That is why in PBL, when I am a facilitator, I see what is being taught by me. They are telling, no doubt, but they bring a diverse references and resources from internet, YouTube, Google, and AI, because they are going to generate a quick answer from the AI. AI fifth generation has come. Now, if you are paid member, you will get more research in that, right? So the students are going to use all these resources and prepare and answer. That is going to be more rewarding than a lecture alone, what I felt. Because sometimes even I was confused whether this is correct or not, because they bring diverse references. So that is going to help. So both are learners here, the not only the student, but also the teacher. So we require a blended teaching, which is going to fulfill everything. Thank you.
Thank you, everyone. I think we had a very good interactive audience. So, just to summarize, I think the organizers are behind us to wind it off. In short, I'll summarize how we moved on from Blackboard to broadband. So, Dr. Utpal sir emphasized on the use of Blackboard advantages and disadvantages, and also the relevance of animal experiments, especially for postgraduates. And Dr. Raichur sir shared the key points which we need to think when we are preparing PPTs as well as presenting PPTs. And also, sir shared his own personal experience of teaching problem-based learning, that how his role by sage on stage to guide by the side. So how he changed himself, adapted to the PBL teaching. And Dr. Anita, who shared that experience of blended learning and different innovative teaching methods. And also she throwed a light on the augmented reality and virtual reality, how they are relevant and how they are not so authentic. And then we moved on to Dr. Vittal, who shared the different online platforms as well as his own experience of running a YouTube channel and how he modified his the videos based on the student feedbacks and the student requirements. And we had a lot of discussion, I think, Dr. Richa Gupta. So she had the maximum questions as she had a first-hand experience of building and learning management system. And we are here with Blackboard to broadband. To summarize, I think what Dr. Anita said was 80-20 principle. So the teacher cannot be replaced by any technology. I think and just we'll wind up with this. So I thank all my panel experts as well as the very active, interactive audience who made this a real panel discussion, I should say. Thank you, everyone.
Thank you, ma'am. Now I request Deepthi Karandi, ma'am, to please hand over the token of honor to the panelists. I now request Dr. Vijaya, ma'am, Professor, Department of Physiology, JNMC, to kindly hand over the token of honor to Dr. Deepthi Karandi, ma'am.
They want me also
Thank you, ma'am. We can now disperse for a tea break. We'll be meeting after 10 minutes.
Hello? Hello? अभी इसका कैसा? हेलो? आफ्टर दिस हेलो? हेलो?
Video stop. Input tomorrow.
हाँ उनका कैसे होगा?
How we are.
हेलो? हेलो? हाँ दो बरस का है। 2 years का हाँ सर 2 बरस का मतलब हाँ 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 सर दीजिए नहीं कुछ मत निकाल
ये आपने डाला था? मैं उनका डाला था नरेंद्र कुमार को.
Yes. But
We welcome you all for the invited lecture sessions. I welcome our chairpersons, Dr. Hanjabam Barun Sharma sir, faculty in charge and associate professor, sports and exercise medicine and sciences, lifestyle and performance medicine, Department of Physiology, Institute of Medical Sciences of Banaras Hindu University, Varanasi. I also call upon Dr. Gagan from AIMS Patna. Sorry, sir. They'll be chairing the session. Sir, I request you to introduce the speaker.
Thank you, madam, for giving me this opportunity. The first speaker of this session is Dr. Narendra Kumar Yadav. Dr. Narendra Kumar is the first FRI civil servant appointed as the Fit India ambassador. And the topic is about Fit India movement, igniting a nation fitness culture. So I invite Dr. Narendra Kumar Yadav. Okay.
Thank you so much, and very good afternoon to all of you. So, already sir has introduced me, but still the theme is make India fit and healthy. So already I was listening, lot of talk was going on, but the perspective of Fit India, I want to tell you, but the people I'm seeing before me, the audience, I think all are faculty members, and you people are having more experience than me in medical field. As ambassador, I'm the first ambassador who is non-athlete. First time government has appointed a civil servant in Fit India as a brand ambassador. Because the government thinks that fitness is should not be limited to only athletes. It is beyond that. And when this campaign Fit India movement was started in 2019, it was well thought that fitness should be integral part of our daily life. So the purpose is to make our India fit and healthy, and to making our country fit and healthy, we need a strong base of healthy citizens. So how Fit India is playing its important role to make our citizens fit and healthy, we'll discuss in coming slides. And also I will seek your suggestions how we can improve this movement. So what we are doing in Fit India, let's discuss a bit. I am sure that you have some point of time have seen the Fit India application, and might have participated some activity as well. So if we see, there is a Sunday on cycle. If you see, one of the signature. Do you have mic and for mic? Restricting. So Sunday on Cycle is our signature program in Fit India movement. It's also kind of symbolic program. Every Sunday across the country, we conduct this cycling to showcase and promote the cycling as a fitness tool, as well as a healthy medium of fitness. So cycling is very important, as I was talking to Harpreet madam. She is do ultra cycling beyond 500 kilometers. That's great. So cycling is also very important mode of to keep yourself fit and also environment friendly. And other programs are also like Freedom Run, Fit India Quiz. Fit India Quiz is very important for the students who are in schools to engage the students uh, by asking quiz about the fitness and health. So these are the few statistics, how we are getting and gaining momentum. And let's go ahead. Statistics don't tell much. So how the basic idea is that how we can make fitness an integral part of in our daily life. If we see in our country, we can categorize the people in majorly two parts. Who are up to the age of 21 years. We can catch them in schools, college, and in universities. Let's simplify the fitness because if we want to make India fit and healthy, so we have to cater every citizen. And if you see in our country, most of the people who are beyond 21-year age, they are facing a lot of non-communicable diseases, challenges like obesity, diabetes, and diabetes, and BP. So, and if we see the students and who are going to schools, colleges, they are bit active, and moreover, as per the data if we see recently, the students in India in last one decade, the activity level has gone 50% down, and the increase in screen time has doubled. It means that our schools and colleges are also not focusing on the active activities in fitness or sports. So now we need to focus on the our schools, colleges, and universities. So Fit India, from Fit India, what we are doing, we are accrediting the schools, colleges, and universities as Fit India colleges, Fit India accredited colleges, universities, and schools. And there are certain norms the school should follow if they are Fit India accredited. And also we are trying to see nowadays we are having a very beautiful dietary guidelines which has been made by the National Institute of Nutrition in coordination with ICMR. But I think most of the people are not aware about these dietary guidelines. And even if you will talk to common citizens, they are also not aware in universities who are studying nutrition, only they know. So what we are planning from Fit India, because this book is very authentic and it is outcome of the research. So we are telling to the schools, colleges, and university, you make the small videos, reels, because Now Gen Z is on social media and on reels. So if there is any sports activity or sports class, you play that reels or video so that a student or a man uh, who is seeing this reels in social media or in schools, they can learn something because no one is there to teach them. So Fit India is taking this responsibility by this way. Also, we are proposing one more important thing, like mark sheet or report card. Fit India is proposing a health card also of every students in schools, colleges, and university. Every year, if suppose we are getting like Chinmay sir was asking about the data, we are also will have in our hand the raw data of our students, our universities, how fit they are, so that if we are making any policies related to health, we can use that data. And if we will catch the our generation at early age, suppose in schools and colleges, then we can secure our future as well. Because in coming days, they are going to take charge of the country. So if they will imbibe the importance of fitness and health, definitely they will follow that in future as well. Now, the most challenging part is who are beyond 21 years of age. Now they have come out from the schools, universities, and colleges, and they are busy with the jobs, are struggling to get a job, or they have joined some corporates, or they are working with the governments. So most challenging part is to motivate these people for fitness. If we really want to improve the health of our country, we have to improve the health of this age group at least. So, what are the ideas? If I ask you people, because when the point come for the fitness, we simply avoid or ignore it because we don't see any immediate problems. If I want to do exercise today, nothing will happen to me. But if I want to go office for a day, I may get a memo. So people, if don't see immediate problem or immediate reaction, they don't bother. It's a common psychology. So to keep these people motivated, it's a big challenge for us also. Like to make this movement successful, these people are a bigger hurdle, or to keep these peoples on track. It's a really challenging because up to this age they have developed all their resistance, kind of resistance, and they think that they are more smarter than government or any suggestions given by the others. So we are proposing few ideas. I also want suggestions from your side, because if we go deep down in the problems, we find that people are give the excuse that I don't have times because I'm too busy. I have to do project late night, and I have to attend office in early in morning. So we are busy. We don't have time for the fitness. So what we are proposing here, okay, you are busy, no problem. If you have downloaded the Fit India application, so what we are planning, we are giving them motivation. Suppose if you will do exercise for 90 days, then you upload the selfie for 90 days. If you are missing 4 5 days, okay, but if you are more than 80 days your attendance, you may get a chance to meet a sports minister, and you will get a recognition. So it may work because people want recognition, and this is the major problem. Suppose if you are in your society and you are recognized by the sports minister for your fitness activity, and then your आपके जो पड़ोसी होंगे, they will think that we should also try to lose our weight or try to get in shape, so that next time may I get a chance to get some recognition from minister or get some award or reward. The basic idea is that we want to motivate the people, and the other idea what we are bringing in to motivate these people, we want to start like a employee of the in private sector, in performer of the month or performer of the week. Now we are planning to bring like a employee of fittest employee of the week. And if you may get also recognized, like if you say good governance award, now you will in coming days you will see fitness and fittest employee of the award, or if you have contributed in fitness, you may get recognized by national level. So, and also we are planning to place like LCDs there, big screen. If you are getting a award of fittest employee, we'll put your photo pics in that digital screen, so that everyone who is seeing and going, they get some motivation that next time my picture should be there. I will also try to improve in our health. So these are the small ideas from Fit India side we are working. And also in corporate, we have a lot of tie-ups with the corporates, and uh, they are already doing it. But now what we have proposed them to, because in corporate people are most of the time they sit in the chair and they don't move here and there. So they are laying like putting the heighted tables so that they can work means walk and work kind of things, not to fix itself some certain places. So that is also corporates are doing. And the people who are self-employed, what we are proposing for them, because they are not in our control, so we are thinking now, if you see, mostly there is a accessibility barrier also in fitness. Accessibility barrier means half of the people are if you see are women in our country, and half of the male. And if you typically see a life of a woman from day starting of the day, she prepare the breakfast and other things for the children, send the children to schools, and then send the husband to office, and she won't get any time for her fitness. So government is also recognizing the efforts of female, and they are planning, means Fit India is planning to at least give them a accessible point. After that, if they want to do some workout, there should be some weekend plan or week plan thrice. So we are trying to come out some ideas, so we can give accessibility to these class also, which where they are not getting any place. And other thing, if we talk about like regulating, this is also very important, because in India, if you see, everybody is fitness trainer, everybody is nutritionist, and everybody is suggesting anything on social media. There is no standardization. So what we are planning to standardize the gyms, all the fitness center. Now, Fit India, what we are going to do, we will prepare a certain questionnaires. Suppose you have done certification of your nutrition or personal training from any institute. Irrespective of that, you have to clear the Fit India exam. If you will clear that, only then you will get the license to work as a fitness trainer or work as a nutritionist. Because it is very important nowadays, casualty are taking place in the gyms, and sometimes those casualty happens only bad management or bad training by the trainers. So we are taking these thing also in our cognizance and working on that. So the ecosystem in India, if you see, most of the gyms are unregulated and they are not tidy. So government is also thinking that to standardize all the gyms, not only by trainers or nutritionists, but also certain norms should be there for the gyms to keep the hygiene well in the gym. And also we have seen a big pandemic a few years back. So the most impacted industry was this fitness industry. Government is also planning, suppose if you are running a fitness gym or yoga or something, so certain licensing will be given by the government, a certain license fees you have to pay. And in lieu of that, in future, if any pandemic or any such conditions happen, you will be taken care by government, like a insurance kind of things. So that because now this industry is growing and it is completely unregulated. So Fit India, we are planning that also. And online fitness courses, as I told in beginning, the all the dietary guidelines and other things of governments time to time will be posted on social media and other mode. Mission Fit Yuva. So if we see our youth, like WHO, 70% children, school going children are not meeting the 60 minutes of daily physical activity. It's a very alarming situation, 70%. So, and if you see some statistics, it's a very important ones. You should definitely read it out, because less than 30% of children are able to meet minimal fitness standard. Means nowadays only all the youth, they are spending their much time on screen. They are not spending times on ground or not involved in physical activity. If you see nutritional deficiency, vitamin D and iron, this is also increasing day by day in our country. Like already I discussed, ICMR guidelines are people are not aware, so we will make them aware, and awareness about the protocol of Fit India and fitness. We are doing it to giving them accreditation and organizing the fitness carnival and different kind of fitness activities. And also one very important thing, we are planning to connect all the boards, schools, universities through API. Suppose if we want to tell anything to the universities, colleges, we can transmit immediately in single click. It will connect all the school college through API. So we can transmit the any information, any data immediately, and it is very much needed nowadays. Everything is going on API and simplified. So it is going to help Fit India and this movement to take it further in a simpler way. Already discussed. So Fit India means what is our approach? We are going to do it in phase wise, and our target is to connect all the universities, whether it is private, it's government, not only government, but we are going to start with the government ones, because that will be easy for getting the data and manage. Yeah, some statistics are here very important. If you see the diabetic, out of 140 crore, 21 crore people are diabetic. We are going to be the diabetic capital. 21 crore is a huge number. And this is only most of big reason is non-active lifestyle. And if we see the BP, hypertension, 22 crore. So if we really want to contribute to make our India fit and healthy, so we have to imbibe the active lifestyle in our daily routine, irrespective of the.
Five more minutes, sir.
Yeah.
Sir, 5 more minutes.
Yeah, I'm binding it up. So these statistics are very alarming for our country's health, and we need to focus, and we need to take responsibility. Suppose if every individual take responsibility of their households, तो हमारे लिए बहुत आसान हो जाएगा अपने देश की हेल्थ को इम्प्रूव करने के लिए. These are the means, as I told, 21 plus age group. They are big challenge. So we can categorize government employee, private sector, self-employed professionals, and we are focusing on these people because they are also increasing the cost of health infrastructure. And if diabetic and obesity will keep on increasing, the spending on budget on health we have to increase. So these are the some ideas we are planning from Fit India side, like mindfulness Monday, tidy Tuesday, wellness Wednesday, some kind of activity so that we can engage the people every day through any media like social media or physical, like 90 days habit. The very important thing is that if you are doing anything for 90 days, then it will be integral part of your life. You need not to be very further, because once it will come in your habit, then if you are getting recognition or not, it will not make any sense. You will keep on doing by your own motivation. So to start to break the inertia is important. The Newton's first law, like inertia, we are stuck with that. We have to break that only. For that, we need some force. And if you are putting it for 90 days, then it will be automatic. Like recognition, motivation. We are also planning celebrity meetups, because sometimes people feel that if they will get the chance to click the photographs with some celebrity, and for that they can engage themselves for doing some workout. These are the just ideas for motivating, to motivate the people. And the basic motto of fitness ki dose आधा घंटा रोज, but rather I say we are having 24 hours, at least 24 minutes in a day. 24 minutes if we are doing workout, and not it will improve the health of ours, but also the health of our country. So 24 minutes we can take out from our busy schedule. Thank you. Thank you so much. Any questions?
Anyone wanted to ask any question, please raise your hand. Please ask any question if you have.
How was the ideas?
Mike.
Already we have made the policies, and we are giving the certification to school. So
But, in practical thing, none of the schools have that, like, is there any registration is cancelled if they do not have
slot
Correct.
for.
You want to tell that if they are not following the
Yes.
terms and condition,
Yes.
so we are taking any action against them or not? You are want to ask this question?
Sorry?
From Fit India side, what is your
No,
question?
within a side, in case, okay, there is policies are made, they have to have a.
They have to
curriculum
follow certain
1 hour.
protocol, yeah.
1 hour. Okay, so in case if they do not follow that very strictly, is there any monitoring on, then if they don't follow that, then what is the next?
We are closely monitoring. If they are not, yeah, I'm telling to you, we are having ambassadors also, Fit India ambassadors, and also what they are sending the reports to us. If they are conducting any, suppose we have given them protocol, you have to this, you have to do this. If they are not doing and doing, they have to submit the report to Fit India. So they are submitting the reports on regular basis. If they are not submitting, our ambassadors are visiting the schools and all. So they are telling them, you do this, you do that.
Okay, so statewide, okay, I know it is not practically not possible to reach out to every school, but if there is anything assignment given to every taluk wise, there is one Fit India ambassador who takes care of that particular
We are
taluk.
planning.
If you
it.
do
we are,
that,
we
I
are
think-
we are planning on this to actually now Fit India ambassadors are only, if you say if they are doing, it's okay. We have not made any fixed responsibility. Suppose these are the schools you have to take care. This is your responsibility. It is not like that. It's only optional, volunteer. So we are planning also in future to make it mandatory for them.
I ask? Time, hai na? This is related to what we talk about, the motivating people, making them motivated. A lot of effort are going on, and we're really happy to see that regarding awareness level also for us. But what I see regarding the fitness behavior, environment is major concern. Now, I'm not talking about pollution. I'm talking about our surroundings, the food choices available, the advertisement are doing their job. Okay, like you have the now cigarette with a cancer thing. Can we have something on the food levels that?
Very good
So
question, sir.
we are not modifying the environment, and we are just modifying the people perception. But we are there. Fit India interaction will be 30 minutes a week, and the other time they are totally bogged down with the advertisement that are so deceptive, and you fall prey.
That's a very good question. That's why if you have listened me carefully, I discussed about to making the reels, means what is the good food, what choices you should make, which are having more salt, which are having less salt. So, certain things are there, and we are planning to create the content on that and play it in the schools as well as on social media. So that suppose if you are getting bogged down and coming across by these reels, your subconscious mind will notice it. So the choices you are making right now, right away, it may change. So we are planning to throw the knowledge. How
But
much
what
we
is
can.
what my concern was like this is about the choices that person can make and making aware the, but at regulatory level I'm talking about availability of such food, regulating the composition of that food, contents of that food. We see a lot of content
It's
now
a
related.
FASI lookout, not a Fit India
That,
point of.
that integration, I'm saying
Yeah.
it will be their lookout only. But if you talk about that level of integration, then commercial thing will it will go away. Matlab,
I got your point. Definitely, I will tell to the senior officials.
Why we always ask that it should come from authority. Right now we are in the APPI conference. So I think either APPI as an association or department as a physiology, we should take a lead that from each physiology department, let's have one fit ambassador. And I
Right,
request you
Chilpa sir.
that from means as your ambassador or you are getting data from the colleges, I request you to include medical colleges now, because if medical college people are fit, then they will guide all other people and all other colleges to fit.
100%, we have to take their own responsibility. If we are aware and we are talking on the fitness, so first responsibility to make ourselves fit.
Physiology means our natural body. So I think it is our responsibility. They are supporting us. So let's, as a physiology, we take this as an opportunity.
Correct.
And we take help from it. Thank you very much. Excellent idea you have suggested. We will try to incorporate it.
Thank you, sir. Thank you.
Thank you, sir. Thank you very much.
It's very fortunate that we have chairperson for the expert committee for the development of fitness protocol, the Professor K.K. Deepak. Sir, if you want to say something.
Yes. So thank you so much. We formulated the protocols and guidelines and they have been published. I hope you all of you have seen it. Now, what has been transpired, we as a mission, we have produced the guidelines. Now, they can be taken as a research label by the physiologist. You apply to the whole campus or institute and see how do you change the fitness level in the 6 months. Our protocols are they're free of cost. We made it sure that no cost is involved except the weighing machine and some other for BMI. And for both quantification of your fitness and also increasing your fitness level. So all 5 components of fitness, flexibility, agility, and all of the endurance have been included. And so beautiful protocol about 30 minutes. And now it's onus with individual and the society and the college and other thing. Nobody stops for APPI to take up this protocol, find out how many of the members here are fit this year and next year. Somebody can do research even on by Google or KLE University or other universities, apply on the campus and see how productivity, how fitness, how the outcome parameter. Somebody has to take kind of a research, protocols are ready. Now, one day show is okay, fine by minister, another thing, that's okay. One day talk is also fine once in a while. So that's an awareness. But what goes to the fitness every day, it's your this thing. So I think I would suggest organizations and individuals to measure their own fitness. And don't compare what is the normal. Like people come, what is the normal? I said, no, we are not defining the normal. We have tried to define normal for children. 10,000 have been done. So those data is there. One of my colleagues, Sujit Parnagrahi, who made this app 365, has some data set. They have the. It's very difficult to define the normal country like us. So we are not competing with any of the benchmark. You compete with yourself. Today you measure, and our protocol can give you numbers. I can suggest you for 5 years to 100 years, it can give you numbers for agility, for endurance, and it has a specific age-wise protocols. Even young age, we have broken into 2. So my humble suggestion, take into an application mode, research mode, and publication mode. Maybe some group or some other takes it, up to you. So thank you. Thank you. That's my comment.
Thank you so much, sir.
Thank you, sir. I now request our chairpersons to kindly hand over the certificate of appreciation and the memento to Dr. Narendra Kumar Yadav, sir. Thank you, sir. Moving ahead with our next topic, cardiovascular health in the era of exercise. I kindly request our chairpersons to introduce the speaker.
Next speaker is Professor Sokumar Jarial. Sir is one of the doyens in the clinical physiology. We all have learned under sir, and we are learning under him. And sir is so humble that he is kind enough to permit us as a chairperson. And I also thankful to the Madam the Harpreet Kaur to allow, to give this once in a lifetime opportunity. So thank you very much. Professor Ashok Kumar Jaria.
Thank you, everyone. So when Harpreet asked me to speak in this fitness idea, so I told Harpreet, I don't do research in this field, so I don't have my own data to present. What should I do? So, but I said I do exercise and I do prepare for things. So can I use that understanding? So I'll give you the background as to why I got into this. I used to do exercise once in a while. That was my not a regular exercises, but I started doing this kind of stuff. Started climbing mountains. And this I started about a few years back. So this is taken from one mountain, and those are the peaks. I went to see those peaks in one go. That was the goal that time. And as a physiologist, teaching everything, so I was trying to look at what problem I'm going to face there. So one my thing was hypoxia, obviously, because at these heights, the barometric pressures are about less than half. So even the PO2, which is 160 here, maybe 80 or 70 there. So for one is to stand, and other is to walk there and climb there. So I thought of ways to how can I mitigate this problem. So after reading a lot, I understood that one of the ways to mitigate this problem is I have to increase my cardiac output. I have to raise that level so that even less amount of oxygen can go faster. So, so just to, so I'll just run through a small video to pep up the area, and then come back to this talk. So I have small video which of the climb. I'll just rush through it. So this was about 20,000 feet height, and so in the night we climbed. Thank you. Okay. So this is the climbing. We got good close, so the temperature is not a problem. And this is near 80-90 degree climb. So of course, with the stones, it's easy because you got a handle and grip. So you really don't have to, but still need the strength to climb and to lift yourself up. So, and that's fortunately your own body weight and whatever you're carrying on your shoulders, which can be about 5 to 10 kilos. So you keep going like this, and so long climb, about a kilometer climb. So you reach this top area, which is the snow area comes in. Now here the things change. Now you got to wear the crampons because you have to put something in the shoe. Now that's another issue which I realized later, that you got to train for carrying heavy shoe. When we train in gyms, our shoes are very nice, soft, lightweight, and featherweight. Now suddenly you got 2 kilos on your feet, and then you realize, okay, the muscles are not functional. And then also the balance goes off. It's like walking on heels. So if you're not used to it, you will trip. And so that problem started coming in. So this was the climb we had to do. So this was the starting point. We went through the area, a long walk, and sorry, it jumped up. Right. Getting stuck. Anyways, so maybe if I could actually show you the long walk, it will be nice, but I'll have to rush through. So back to the talk. So I started preparing for it, and when you go online, you find so much information, and you don't know which one to use. You don't know their background. So ultimately, being a physiologist, I fell back to published papers in physiology. What does the actual data tell? Not what the celebrities, experts, successful people tell. That's separate. Come to the brass tacks. So when started studying, so you look as to evolution is something that we all believe in that. So how is our body created? For what purpose? So we exercise in the history for these reasons. So first soldiers were exercising because they had to be fit for fighting. Then sports came in, adventure sports came in. Over the last few years, bodybuilding, aesthetics, you want to look good, aesthetics, not necessarily driven by fitness. You want to look good, which of course fitness will come with it. And lastly, of course, the major group is mitigation for adverse effects of our lives, diabetes, hypertension. So we are going to go about this. So I'm not going to talk about how exercise affects these. I'll fix on physiology. So in the context of human evolution, humans evolved to do what is called as long distance endurance running. We can outrun anything. A tiger may run faster than us for some minutes, but eventually we'll catch it. Because we are the only animal which have a fantastic thermoregulatory control. All other animals will fail because the temperature goes so high, they just cannot maintain it. So high speed, how long? Okay, only some of the canines can outrun us. Okay, or the kind of horses. Otherwise, most animals will catch. And largely it is because of bipedalism. Our efficiency is very good. Excellent efficiency to do the same covering the same length of ground, humans are the best. We have the most efficient system in walking. When we go high speed running, for which we are not built, we're not built to catch tigers and deers in one go. We are built to catch them over a period of time. So we'll keep running, and they'll keep running fast, so, but until they get tired. Because they don't have the kind of reserve. We have a little more fat than they have. So we will be able to catch them. They don't have water understanding. So we evolved for this purpose. So human walking is allometrically most efficient. So when I hear people that we are walking every day 1 kilometer or 1 hour to lose weight, I'm saying you're using the most fuel efficient mechanism to lose fuel. It won't help you. If you have to lose weight, then you have to go slightly different. You have to go to high speed running. Then allometric, when I use allometrically for a given body weight across mammals. So we are the most efficient. Okay. So because this has effect on how your cardiovascular system has evolved or adapted. And therefore, when we push it, we should know what it's going to do. So if you're walking, we can walk for hours and hours. We can cross a huge distance. But high speed running, you won't be able to do. Middle length running, about 50% of your VO2 max, you can run for long. Marathon running is 50% of VO2 max. They don't know that fast. The fastest runner, it will be 40 kilometers per hour. Marathon runner is 20 kilometers per hour. The best of us. Now, exercise trends. We go out, you find lots of trends, popular trends. By intensity modality, one can talk of high intensity interval training, cardio, aerobics, strength training, functional training, yoga, Pilates, so all kind of combinations are there. And you'll have people telling about which one is better, which one is not. As a physiologist, I say this is just, let me look at what happens to the body when no matter what you do. So first I'll list the misconception, and then come back to concepts, then back to misconceptions. Popular ones are these: no pain, no gain. Very common. Very common. They'll come back, we are sore, exercise when it is sore. Very common. And largely given by, again, unregulated gym trainers. They don't know, they have learned from hurt or somewhere. Okay, abhi you do, DOMs will come for next 3 days. I've seen so many people start exercising, then they're walking difficulty for next 3-4 days. And say, why? And then say, can I walk? Even my gym is telling that you walk now when you're going to damage yourself. Spot reduction of fat, again, very popular these days. You want to lose arm fat, do this. Leg fat, do it. Doesn't. Muscle changes to fat if one doesn't exercise. Again, very common. More sweat, better workout. Okay. Cardio is best for losing weight. In my understanding, depends upon the rate at which you're doing cardio. If you're doing a slow cardio, you're using the most fuel efficient system to lose fuel. Exercise can offset poor diet. Again, very, very common reason given for eating bad food. We will eat, then we'll exercise. Okay. A high intensity or too much cardio is bad for heart. If you got a heart problem, don't exercise. We are born with finite number of heartbeats. If we exercise, heart slows down, therefore we live longer. Okay, ये भी misconception चल रहे हैं. Exercise for heart need not start at young age. That only when you're 40, 50, then you start. Otherwise, heart is young. And other problem is duration and days of exercise. How much should you do? Minimum. So then somebody says do every day. So I'm going to put this in the evolutionary concept. Okay, humans never did run, but didn't have to run every day. Sometimes they would run, sometimes they would not run. But that was the time when our surroundings were different. So what we're doing is largely mitigating for the problem that we've created. We did not evolve for high calorie, high salt food. We didn't evolve for diabetes or other stuff. We didn't evolve for our food and sensory habits. But now, since we are using it, now we are mitigating it. So we created a problem for us, and now we're going to do it. Okay. So I'll go to pure physiology concepts. There is a community clinical data. I won't touch that. At which exercises is good for this heart or that. I'll purely touch on physiological data. So I went through a huge number of original papers, for thanks to the net that you get all these papers these days. So things that we know, but these are quantitative data. Our textbooks of physiology don't give quantitative data. This all graphs I've made from all these references. This is quantitative data, which means the wattage is given below, and this is endurance running, not isometric exercise. So oxygen uptake can go up to 4 liters, which is pretty high. From 250 ml, 0.25 to 4 liters, and This is the wattage which maximum most of us will be able to go. Now, if you look at 4, 2 would be your 50% VO2 max. Given the, of course, we have individual VO2 maxes, but for a person, it will be around 2. For a healthy fit person, it will be around 2 liters per minute. So this is very important when we discuss later. So cardiac output also increases. The data shows about 30. Okay. But this is again for a fit person. Unfit will reach to 20-25 liters. Now, heart rate, same linearity. What interesting is the data on the stroke volume. We have different kind of people. Some people have linear increase, B type. Some people have A type plateau. Some people have initial increase, plateau, and then increase. And some people have D type. This is depending upon your heart condition. So D type would be those in early failure. That means they are grade 1. They're normally healthy. But when they increase the cut to that level, the heart cannot pump enough now. So these 4 patterns are in the data. AVO2 difference, of course, linearity. More again, if you pay attention to the BP data, and this is something which I find very funny when we have practical examinations, and the student is asked to perform a effect of exercise on blood pressure, and they all say fantastic blood pressure rises. So this is the real data, measure with intra-arterial pressures and cuff pressures. You see diastolic pressure? It hardly changes. In fact, rise of BP is deleterious to heart. It increases the workload on the heart. Actually, BP must not change. We were built for up to 1.5 or 1.2, which a human will actually run on for long distance endurance. So we see that there's a hardly very small rise, 10, 20 BP for up to 2 liters. Thereafter, it begins to rise higher. Because evolutionarily, we were not built to do high speed running. We were built to do 1.5 to 2 liters running, slowly stop, run, stop, and catch the prey. Hunter-gatherers, not otherwise. So something that is important, that diastolic pressure, because it's the reverse thinking that high pressure will push the blood. Actually, the high pressure is because blood is not going there. But there is no battery pushing it. Like when we apply Ohm's law, is applied as there's a potential difference and something resistance. Actually, the pressure is because there is a resistance. Heart is pushing it. So even Ohm's law is incorrectly applied, I think, in physiology as a potential difference driving something. If artery was open, there will no pressure. Pressure comes because of resistance. So this is the pulmonary circulation has a linearity because pulmonary circulation does not have the responsive neural control. It's like system. Now coming to the respiratory system, again data. By 2 liters, you will go into anaerobic metabolism, which again tells you that humans were not designed to do more than 2 liters. Then you won't be able to run. Okay, the acid will begin to. But when the buffering gets compromised, then we go into respiratory compensation point. So now we are breathing much more than. So now it's pH driven. So healthy running for me, it would be less than 2. But don't go higher than that. You are entering into the anaerobic zone. For sports, different matter, but for healthy. On the side, diffusion capacities and both, and this is all taken from all this data. Oxygen uptake, very interesting data. Even before you begin, oxygen uptake has jumped. How was the oxygen uptake jumped? Where is it going from? So we have enough oxygen in the blood. So what happens is the 1.5 liters in the spastic circulation gets shifted to the main circulation. So 500-700 ml cup, which is the splanchnic is a slow flow with low O2. So when it comes up, it picks up oxygen, and therefore there's a sudden rise in the first few seconds. This is very interesting. Oxygen uptake just goes up. First, over 1 minute, the oxygen uptake rises. Okay, that means to stabilize anything, you will need at least 1 minute, no matter what level of exercise you do. If you start running at 5 kilometers per hour, 10 kilometers per hour, whichever rate you want, you will take 1 minute to stabilize. And that is why the protocols for exercise testing always have at least 2 to 3 minutes of stabilization. And there's a step. That step, 3 minutes, step, 3 minutes. You can't go less than that. You should not go less than that. Then, again, interesting data. The bicarbonate level begins to change because the buffering action starts. Obviously, that's why the anaerobic threshold begins. In the blood, there is no norepinephrine, epinephrine. Everything is within the neurons. It doesn't come in the blood. Only after 2 it begins to come in the blood. So adrenaline release of sympathetic is going to come later, not early. So again, my understanding from the physiological data is that anything above your 50% VO2 max is not for which you evolved. We have to, and that's the best marathon runners run at 20 kilometers per hour, not 40 kilometers per hour. So now, physiologists, some concepts. You may not answer this to me, but in your mind, what is the mechanism of increase in ventilation during exercise? That's the common question we ask students in the class. Now, this is the correct data that PaO2 and PaCO2 does not change during exercise at all. Then the question is, that's what we teach. Okay, oxygen demand, oxygen fall, trigger, carbon dioxide rise, trigger. It's wrong. Completely wrong. This is the data from 100 years, from J.B. Haldane. Still it is correct. In fact, at higher intensities, PCO2 drops because of rise in potassium. Potassium stimulates the same chemoreceptors. So we are now hyperventilating. I showed you this graph. Exercise, hyperventilation, top right. Now you are ventilating more than the production of carbon dioxide. So we confuse between production of carbon dioxide versus level of carbon dioxide. And this is common. Unfortunately, it has sort of reached a Chinese whisper that from what it started and what it has become. So this is the correct thing. That then what makes it hyperventilate is the next question. If this is not the reason, then what is the reason? Next question that, so people tend to answer why of question, not how of question. They say oxygen is needed. That's a why, not how. Physiology is about how. Why does it rain? How does it rain? Two things. In language, we say kyun hua or kaise hua in Hindi. And we have a particular word. In English, we confuse the two. You are asking, so we should never ask a question of why does cardiac output? We should say, how does cardiac output increase? But because English is not our native language, we tend to use them interchangeably. We'll never do this in our native language. We'll ask exact question. Kyun karte ho aur kaise karte ho? It's because I'm in Hindi. But in English, we tend to mix. And the student also then give the answer, mixed answers. So we have to focus on whys of things. For example, somebody says, why does it rain? I say, just to make me happy. That's why it rains. Why can be anything? How does it rain? There's only one answer. Water cycle. Let's go that way. So, so this is the timelines. Okay, all these. So what is the mechanism of this rise? In 1 minute. So I'll quickly take you through some data. Increase in venous return, stroke volume, cardiac output occurs at the onset of exercise. You don't even have to run. It'll just start doing its job. Okay. So it has to be driven from top and from periphery. It is not what we call as homeostatically driven. Okay. So mechanism of response, this is the typical diagram that we show of the negative feedback mechanism. I've laid down all the inputs and outputs to the system. This is a very detailed diagram. And top down also the detailed diagram. These have all been built by the information available. And so understanding is this. I think this is slide if you physiologists can hold up. So centers of behavior. When you have to behave, which running is a behavior, catching somebody is a behavior. That's what we evolved for. So there is central command, which comes from that is estimate of intensity. How much you're going to run? The system already knows. Give feedback to the central centers, which is medullary centers, below. Muscle activities, what is actually happening? So I may not have yet run. System will pick that also. So it needs both the things. It needs how much you're planning to run and how much you're running. And they can exchange for each other. It's not additive. The data is there that even if you move passively legs, BP and everything changes. Heart rate changes, passive movement savvy, because the system is coming. All you need is tension. So there is tension in the tendons and the muscle. It goes up, it stimulates the right areas. Then there is cardiorespiratory change, ventilation, perfusion. If there is a mismatch between the expected and what is happening in the cardiovascular, only then the reflex mechanism comes into picture. Only for fine tuning. So negative feedback on the left side is only for fine tuning. Actual driven is top down and bottom up to the medulla, and then it does. So this is something that if it is good. Now, from a cardiovascular perspective, ultimate is coronary blood flow for most of us. That's where the patient land up in problem or the ischemia occurs. There's a complete correlation. I'm not talking about causation. Correlation. So we say exercise intensity, proportional will be oxygen demand, proportional would be the or oxygen uptake, cardiac output, heart rate, cardiac work, and coronary blood flow. So cardiac work becomes the central point. What makes the heart work more? One is heart rate, and two work, volume work, pressure work. Pressure work is very difficult. Therefore, rise of BP during exercise is not a good idea. Rise of stroke volume is a better idea because volume work is less than the stroke work. And humans have developed to 2 liters per minute. Beyond that, the pressure work begins to increase. So whether, because HIIT reaches to that level, and therefore it may have difficulty for those who are having heart problems. So another question, what sets the maximum value of oxygen uptake in each one of us? Is it pulmonary system, oxygen diffusion, or transfer, or oxygen carrying capacity, or how much can I run? All four can be the limiters. Data is very clear. It is just the cardiovascular system. How much your blood can go around. Pulmonary system has sufficient capacity. It will never be a trouble. The best of the athletes do not have pulmonary system better than any one of us, but they're better cardiovascular system. Then oxygen carrying capacity, most of, unless you have anemia, we have no problem. Blood doping had been a cheating mechanism for high long distance runners and cyclists because they would take out the blood, put back in to increase the carrying capacity. But it's not a limiter in the regular sense of the word. Of course, skeletal circulation, people thought whether the muscles blood supply to the skeletal was it's not a limiter. What is limiter is cardiac. Next question is, what limits the cardiac output? Okay, so we know that athletes have raised maximal cardiac output and consumption. We know that they have resting heart rate. Again, a question. Why? What makes the athletes have lower heart rate? We say parasympathetic rise. We don't ask the next question. What makes the parasympathetic trigger? What is, because you have to reach the ultimate answer. So our textbooks and everybody stops at the first level. Heart rate is low because parasympathetic tone is high. Very good. Now, but why? What makes it? So again, why and how of the question? Let's go to the how of the question. So again, I dug out a good amount of data that plasma volume starts increasing within few days to about 10% increase in blood volume if you start exercising, running, not isometric. The RBC volume then follows because the kidney senses the hemodilution, erythropoietin gets secreted. So within 3 weeks, your blood volume will go up by 10%. It's a huge value. Then increase in stroke volume occurs because of better diastolic filling, faster. Because the heart has to have nice dilation. The lucitropic effect of the sympathetic stimulation. Beautiful dilates. Very interesting data of 2014. Athletes who have higher maximal cardiac output, just take out some blood from them, they lose the whole ability. That means having a higher, stronger heart, lower heart rate, all are permissive. They are not the cause of cardiac output high. The cause of high cardiac output actually increasing blood volume. If your blood volume is not going to increase, then no matter how strong your heart is, no matter how slow your heart rate, it won't help. With the increased blood volume, you do need heart to pump it. But heart is not the reason. The reason is blood volume. And therefore, data of natural gifted people, some people are naturally gifted. They find they have just a resting higher blood volume. Naturally, they have. So the keys. Now, why this matters? When you give somebody exercise training, dehydration is a major problem. And the first thing you'll get dehydration going to affect is the intravascular blood volume. So coming back to this last slide where I was going to go back is, sorry, I missed it. I pressed the wrong button. So I was on the misconception slide. So let's answer these. No pain, no gain, wrong. Hurt tissues, you have to give them rest. In COVID, the reason why we gave oxygen at 90%, where we teach students at 90% saturation, at 90% is nothing. We have 20 ml in arterial blood, 15 comes back. A buffer of 15 ml. But still at 10% drop, we gave people oxygen. Oxygen was not given to take care of oxygen problem. It was given to take care of lung expansion because you wanted to prevent lung from working. It is given to what is called a self-inflicted lung injury. Even ventilators, you don't want to give too much of expansion. So oxygen in COVID was not given to take care of oxygen problem. There was no oxygen problem. So when they say happy hypoxia, as a physiologist, I say, you will be happy up till 80. Only at 70 will you be unhappy. So as a physiologist, if at 85 somebody is having distress, something else. It's not low oxygen. Hypoxemia versus hypoxia. We will have hypoxia only at 70%, not at 80, 90. Hypoxemia, it's not a problem. You will never land in cellular acidemia at 80, 95. Therefore, only that may come a small bit at 80 is high cognitive function. Other than that, nothing happens. Nothing happens. So when say happy hypoxia, I say, first of all, both terms are incorrect. Patient is not in hypoxia, patient is in hypoxemia, not yet in hypoxia. And happy, you're supposed to be happy. In fact, you're unhappy, then I would be troubled. It's something else. Okay. So no pain, no gain, incorrect. Tissue needs rest. Micro trauma occurs during exercises, you must give it time. Spot reduction of fat doesn't work. There is no specific reason. You have to decrease fat everywhere. And don't target it. There's a study of people in 70s, 80s, and 90s. They're performing 3 decades. The only factor which was somebody directly correlated is little bit of fat. Should have some amount of fat at old age. Don't be too lean and thin. So it's good for, that's the data. Muscle change to fat, doesn't happen. What may happen is just a bit of tissue may occur, but fat doesn't become fat. Okay. More sweat, if you're sweating more, then your environment either is different or you're wearing some different kind of clothes or aeration is bad. Okay. So don't try to sweat and think that you're being better. Cardio is best for losing weight. Depends at the rate at which you're running. If you're going very slow, it won't help. You're using the most efficient mechanism to lose weight. Very bad. Only when you're going to high speed, perhaps then you won't be able to maintain. Many of us won't be able to maintain high speeds. HIIT speed, we can't run for, just try and run 10 kilometers per hour. It's very difficult for many of us. Only the 20s, 30s can run. So if you're running at 5 kilometers per hour, small jogging, nothing is going to happen. You are using the most efficient system. Exercise can offset poor diet, not at all. Do exercise for fitness, control diet for weight. Thinking that by exercising, you will control your weight. And the other issue is that people think, so when you start exercising, you build muscles, weight increases. When you're exercising, the bone mineral density increases, weight increases. So actually, psychologically, your weight is not decreasing on the scale. So what you may be dropping a little bit of fat, but you are increasing the bone mass and the muscle mass. So weight, that may not. So the circumference is the right method instead of weight. BMIs have been taken off from the definition of metabolic syndrome. It's the girth. So I think the girth is the right way to go even for losing weight rather than actually BMIs. It will be incorrect. High intensity, bad for the heart. HIIT, it's not. Unless you got a cardiac problem. Otherwise, it's not a problem. Because you can't run for that long in HIIT. You can only for a few minutes. Then if you have a heart problem, don't exercise. Actually, please exercise, you got a heart problem. Only first, do it in front of doctor. Get yourself a TMT done. Know your levels of exercise. Because you have to push the heart, but not over push it. Okay, and your levels could be 7 met, 10 met, 15 met, whatever it is. You can't practice in the public domain suddenly, and suddenly you develop ischemia. Who will take you? So those who are having a heart problem, best go to TMT lab where everything is there. Start doing slowly. Grade up 1, 2 minutes. Slow. Not necessarily do the Bruce protocol. Some protocol. Find your tuning and exercise at that level. That will be helpful. We are born finite numbers. It's a myth. Doesn't work. Heart need not be started at a young age. We should be start exercising at all ages. The peak health we get is at the age of 30. After that, everything is going to drop. And what by doing exercise and weight training, everything is we are decreasing the slope so that we are so high at 100 that we are healthy. So healthy aging would be better than because we are good dropping. Everybody's duration of exercise. Every bout of exercise is useful. If you because it's the larger barrier. अगर मैं 4 दिन नहीं कर सकता हूं तो फिर मैं कैसे करूंगा? मुझे 4 दिन नहीं हो गया, then you stop doing it because you feel you failed. You started with a tag of I will do 3 days a week, and you could not do 3 days a week. You say, oh, I failed. Then you lose your momentum. Our suggestion is every bout of exercising, no matter where, is helpful. If it is good, if you can reach 3 days, 4 days a week, or half an hour, whichever way. Type of exercise does not matter. It should be, I'm talking only of the aerobic kind, because anaerobic is a different ballgame altogether. So with this, I will stop here and have a discussion query if needed. Thank you.
Thank you so much, sir, for the wonderful talk. Anyone have a query, please ask and your questions. Sir.
But people do talk about if you really want to lose weight, I mean, then you have to put your muscle on work and do strength training.
Yes.
So what is your take on that?
So my understanding is, see, all of us are using a proportion of fat, carbs and fat all the time. When you are resting, as the proportion, you are using much more fat. Very, very, very interesting data. When we are resting, we are using 60 kilocalorie per hour, of which actually 80% is coming from fat, 20 coming from glucose. So बढ़िया पे बैठे रहो. The only problem is it's 60. If you begin to increase, the proportion changes. Now you're burning more of carbs. That's another problem. So your percentage fat burning is changing to carb because muscle need glucose. That's what you're going to use it for. Only heart can utilize largely ketone bodies and fat. It can. The rest of people are now suggesting that what you do is whether you should do cardio first or high intensity first. What they're suggesting is do intensity first. Whatever carb has to go. Now you're left with only fat because carbon is currently utilized. Now when you start running, heart has no option but to utilize. So if you did cardio earlier, heart would have taken glucose as well as fat. Muscle cannot do without. So that's the idea. And recently I saw a paper on this data. One cardio before, exercise later. And the other thing was that if you do good cardio, you may not be motivated to do strength training. But strength training is after cardio, you may just a bit of tired. So strength training is that way not very tiring. Okay, but today's talk was only on the cardio part. So that's what we are built for. He was mentioning earlier, there are so many people coming to orthopedics department with tears, sprains because of heavy weight usage. People are not knowing how much weight to use, how many repetitions, how many sets, how many weights. Again, total incorrect data. Just because every time you do exercise, there is a stimulation to the cells. The two types of growth muscle will have. One is myofibrillar growth, which is strength. Another is myoplasmic growth, which is glycogen. Strength training, so the effect will be both. The genes trigger for 48 hours. If you do any single bout, for 48 hours, gene will be triggered. After that, it won't be triggered. So even a 1 day gap is fantastic. 48 hours minimum, even 72 hours it can go if you are trained well. Right.
Any other questions, please? Dr. Manish.
Sir, in a video you described about cramp. So on cramp, so you told about cramps. You received cramp during in a video.
No, I was talking about the crampons. So there are those spikes shoes. So fortunately I did not get cramps.
Acha, there was different thing.
That's crampon. There's a to walk on the ice, you need 2-inch long, which can go into the foot.
Okay, for stabilization.
Yeah. Otherwise you'll slip. And recently one person died because he just slipped. And once you slip, you can't stop. You're wearing nice cloth, smooth, and the ice becomes water on pressure, and it's like you're skating now. So, because he wanted to take a selfie, so he unhooked from the rope and just went back and slipped and lost.
So my question is, when the exercise person comes, who should be the first doctor who should see with which specialty? And then ranking first, this specialty, then second, third, fourth, and what you rank physiologists in that rank?
Yeah. So we have to differentiate two: exercise training and sports medicine. That's a separate thing. If it is sports medicine, which is sports-related injury medicine, then my suggestion is to a practitioner of sports medicine, not to a physiologist. We're talking about exercise prescription. For a heart patient, again, not to a physiologist, to a cardiologist, because they have experience in it. Healthy people, exercise prescription, I would suggest physiologist, provided physiologists are aware of the real data. See, if our data is on Guyton Genongs and some review articles, you want to have it. Because our books don't talk quantitative, we talk qualitative. भागने से cardiac rate बढ़ता है, कितना, how much time? No, books don't talk, because they are not supposed to talk. That's for people becoming a doctor, not becoming a physiologist. We don't have the high level books for physiologists, empty books. So my suggestion is that for healthy people, it will be good. My suggestion would be physiologist, provided physiology up to that level of understanding and practice. For sports injuries, those who are trained in it, starting with largely orthopedic or those who are in sports medicine.
For the healthy people, we have to rule out the contraindication first. So then physician first, or then or physiologist.
See, if you know the data, your subject well, you will get them tested. So it's, for example, it's a common, not done in India. If you go for adventure sports, they will ask you to get an ECG. They'll have to get a physician's clearance. Okay, and we are MBBS doctors, so we can always provide, if you've not forgotten your doctorate degree, pure MBBS, just examine the patient, gets few tests, should be good enough.
Thank you,
sir.
Arnab, sir.
Yeah.
Oh, sir.
Hello, sir. I had a question.
Oh,
here. Okay. So nowadays you see a lot of fitness enthusiasts, as well as, people, those who are into bodybuilding and other stuff. Okay. They die at a very, very early age. Okay. Of around, 40, 42. So what could be the reason behind it? Is it, because of genetic, predisposition, or is it, because of the wrong, medications, they take? Because, in spite of being so fit, okay, they die suddenly. Okay. So the thing is, I would just like to know the reason behind it, sir.
So my understanding is that even one death in lakhs gets publicized hugely. Yes, even people who have never taken any smoking, drinking gets cancer. That doesn't mean that they're not, you not take care of that. In my understanding, there's nothing to do with exercise. Exercise has been good, it's very helpful, everything. If one or two odd cases occur, there has to be a background arrhythmia, latent, which gets triggered by sympathetic stimulation. So they are not aware of it. It's not causing trouble as of now. When they start exercising, because if you're 30s, 40s, it cannot be, I'm assuming that they are healthy, therefore they don't have coronary problems. They can have. People have family histories where, despite looking healthy, they have heavy coronary depositions, coronary artery disease. They don't know about it. They have no idea. They have never ran. And especially this for our kind of people. See, we belong to a group of people who are highly motivated. That's why we are sitting in doctors and engineers. Motivated, you reach 40s, suddenly you decide to now start exercising, the same mindset begins to work. Now I will start exercising the way I read, the way I did everything. And you gear up, go to there, start running at the best maximum possible. You're going to not slow it down. And so you will be fit and just spike it and land up in trouble. So for me, it is unaccustomed exercise and a background of arrhythmia, somewhere latent. Because I don't think that age of 30, 40 is because of coronary artery problem. If it is, then there has to be family history. Otherwise, no.
Sir, I have one point.
Sir.
I like to know your reflection.
Sir.
What about overtraining?
Sir.
Overtraining and too much exercise without doing proper warm-up. Many a time we see without doing proper warm-up. Warm-up means peripheral vasodilatation so that the sudden surge of blood pressure does not take place.
Sir.
We do the assessment of the footballers of our
yes.
Bengal Football Association,
yes.
Bengal Soccer Association, and we are regularly doing the CPET, etc. Distinctly, there are 4 categories of physiological entity like a defender, a midfielder, and an attacker. They are physiologically different. A midfielder never attains the anaerobic threshold, remains below RER1, still completing the full-fledged Bruce protocol. Now the point is, without doing adequate warm-up, increasing height of activity, which is done by the attackers. They suddenly accelerate, try to score the goal. We find that there are thickening of the before doing the CPET examination with the echocardiography, and the thickening of the ventricular muscle, and maybe that may be the reason many a time, at least once in a year in our state, there is a sudden cardiac death in the midfield, mid of the play. So overtraining and over enthusiastic exercise is rather counterproductive.
It is, sir.
That must be told.
Absolutely, sir. Sir, I'll tell you, just to answer your question and give you background. Pregnancy is associated with hemodilution. Mid-trimester hemodilution. If you ask what is the evolutionary advantage of this kind of change, it seems counterproductive. Mother should be having actually more hemoglobin so that more oxygen transfer. It's actually hemodiluted. And my understanding is this, that hypertrophy is going to be very bad. Heart should not go into hypertrophy, whether for pathological reason or for physiological reasons. So those of us who are pushing the heart to a hypertrophy level will land up in arrhythmia sooner or later, because system was designed for a particular timing. There is a pathway which goes on the inner side, Purkinje. After that, it is muscle fiber to muscle fiber. If you increase the muscle, you increase the time. And the depolarization must occur at the same time. You don't do that, you'll go into circus rhythm. So somebody will get into that circus rhythm, suddenly it get into ventricular fibrillation. And it will take only a few seconds. You won't even know what happened to the person. Because ventricular fibrillation occurs, there is no marker. There's nothing. Or it's just dead. So my understanding is that any exercise for a healthy living should not lead to hypertrophy of the heart. If it's leading, unfortunately, we are pushing people so much for money in sports that they will push themselves to death. And they will end up. So that's my understanding, sir. And I showed you the evolutionary data. You're not supposed to go above 2. You're not supposed to go into anaerobic threshold. But we are, for our livelihood and, of course, glamour. Thank you, sir.
Are there any other questions? Sir.
Just now you mentioned 2 liters per minute, VO2 max. That should be optimum to have a good endurance. Would it be helpful in reducing weight also for weight loss?
Okay. So again, combination is the data suggests combination of resistance exercise along with cardio at a certain rate. Very few of us can run 10 kilometers per hour. Then it will be beneficial. If you're running at 4 or 5 kilometers per hour, your running is not going to drop the weight, because you are using the most efficient system of locomotion. So incline, maybe incline running, because you're still using some resistance, or step ups, that where you use some bit of weight. That would be better than walking, as far as weight losing is concerned. So resistance. Right.
Sir, one more question. We label obesity as a paradox. Sorry,
Yeah.
obesity as main culprit for cardiovascular mortality and morbidity. Now, there is one thing called as obesity paradox. Actually, it tells us that obese people don't die so quickly in the ICU as far as ICU mortality is concerned. They somehow they tend to survive longer.
Yes.
So, what is your take?
Just simple, because see, this is a problem again with diet controls. Glucose kahan se aayega? It will come from protein. Your immune system needs proteins. When system, see, cortisol's one effect is increase glucose. At what cost? Where is that glucose coming from? Not from outside. It's breaking down proteins. And it is shutting down the immune system. So hypoglycemia is the worst enemy. So if you are not having sufficient reserves, which you should be having, when the levels drop, brain needs glucose, it will start burning proteins. If everything else you've lost, finished. So I'm going to take bit of fat, and that's about that 70-80 data I told you. Bit of fat is good. Fat has a big range. Minimum fat is 10% for males. Minimum fat. We need minimum 10% fat. Anything shredded weight is not good. The up to 30 doesn't cause you disease. Up to 30 doesn't. About 30 begins to cause trouble. So range is very large. So my suggestion would be to stay in the upper range of 20, 30s, but not beyond that. But the data is correct. that if you're in ICU, what are we getting? Who's feeding us? The person in 10 days in ICU, what food they get? IV lines, glucose, 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 or you're going to give through. So that food is not sufficient. So then there are your backup reserves. And of all the animals, humans and the other primates have more fat percentage in the body, except the dogs that we keep at home. Otherwise, animals don't have less than 5% fat. All animals, they have less than 5% fat on their body. We've got 10 to 15% evolutionary, so that we could run longer. That was the evolutionary advantage. So we store fat. That's my understanding, that bit of fat is used. It's a paradox maybe for clinicians, but for physiologists, it's not a paradox. Similar to happy hypoxia. It was a paradox for clinician. As a physiologist, was your terms are wrong and concept is wrong. But then they don't listen to us. They are more louder. Thanks.
Thank you very much, sir, for the talk. Because of lack of time, we are not taking any questions now. Dr. Varun, you want to say something?
Very nice, sir. Because understanding physiological principle is very important, and sir has shown all the original paper and original data and everything. 3-4 concepts are touched very beautifully regarding the stroke volume, and that concept is very relevant for the clinical practice. Another is sir has touched about hypoxemia, arterial hypoxemia. In highly athlete people, because of the fatigue of the respiratory, this thing, arterial hypoxia do occurs, respiratory center. And sir is beautifully talk about, the volume expansion. That is also clinical relevant, where we see, endurance athlete coming with hemoglobin concentration less. And when they go to clinician, they just see the biochemical report without any all this thing. And that is not total hemoglobin estimation is not done regularly, only hemoglobin concentration. And that is because of the, dilutional pseudo the anemia, sports anemia. So this kind of thing is physiology is the extreme core. Sports and exercise medicine is just the application of physiology. So physiology people are the first one. This is what I can take. Thank you very much, sir. Thank you very much.
I request our chairpersons to present the certificate of appreciation and a moment of our resource person.
Thank you, sir. Our next topic for the session is sports-specific fitness, elite of general population. I request our chairpersons to kindly introduce the speaker.
The next speaker is Dr. Major Amaya Kagali. He was a team doctor, Rajasthan Royal, Paris Para-Olympic 2024, as well as a chief team physician of India in that the Para-Olympic. And he is a fellow colleague, sports, the physician and sports exercise science expert. So Dr. Kagali, the stage is yours.
Good afternoon, everyone. Thank you, sir, for the introduction. And it's a huge honor for me to be introduced by Dr. Barum, learned quite a few things from him. And I'm really enjoying being in this segment, and especially listening to you, Dr. Ashok. It was fantastic. I agreed with 99% of what you said. Through the course, you'll know what 1% I'm talking about. And of course, Dr. Harpreet for inviting me. So I won't keep you all starving for long, and dive into sport medicine. So this is just the brief layout of how I'll be talking, just the terminologies. MDT is a multidisciplinary team that we work in elite sport, sports training, then we'll take it as it comes. Anyway, so when we talk about anything to do with sport or exercise, we generally use these terms very synonymously. What is physical activity? What is exercise? What is sport? So in what is a game? So in my view, it's that for every sports person, you need to be physically active. But for every physically active person, you don't need to be a sports person. I hope that makes sense. So physical activity is just something that you wake up in the morning, brush your teeth, go for a walk, just that. Risks are a lot lesser because there's no competition. Automatically, when you go just for a walk, your cortisols just don't shoot up for no rhyme or reason unless you have a target in mind. But that only you have 50-over game, T20 cricket, 50 minutes of hockey on the pitch, you're leading or trailing 1-0, so many other factors. So the difference is that we as lay population or recreational people need to differentiate what the elite are doing and not ape them unnecessarily. It's good to be inspired by them, but there's a huge difference. Majorly what Dr. Ashok told, actually he's made my job a lot easier, because with the misconceptions, I agreed with each and every point what he told. No pain, no gain is, pardon my language, absolute garbage. Okay. So anyway, exercise is, again, you're giving it a more refined form of physical activity. You're going into reps, you're going into sets, you're going to the particular time, inclination, speed on the treadmill, speed on the cross trainer, speed on the cycle. So you're giving it a little more structure. And then of course in sport, all what you have done, you've prepared your physical body. You're implying that on a court or a pitch or a ground, add in the rules, colleagues with teammates, if it's an individual sport, what are your timings, how will you perform, and whatever said and done, no matter what the athletes have to say, that the results will take care of itself, they do care about the results. Everybody in competitive sport cares about results, including the support staff. If you win, we all play to win. It's as simple as that. Nobody plays for fun if it's not competition. You ask any IPL team or any countries in the world, or even Usain Bolt to the 10th person who came, everybody plays to win. So how does a proper elite support staff look like? It's not just a few people here and there making up a team. Okay. Fortunately or unfortunately, in elite sport, coach is the king. So no matter how much of textbook knowledge you have, how much you apply the textbook knowledge to your athletes and to the other support staff, it is the coach is team, period. Football, it could be the manager. Cricket, it can be the captain and coach. Hockey, it is again the head coach. So all you need to do is that not come across as too aggressive and too egoistic, that, oh, I'm a doctor, I know all my things, but I'll impose this on you. The next day you'll be kicked out. And that's the common mistake what I see professionals making, not trying to blend in. Knowledge is power, yes, but the application and the understanding of that knowledge, and most importantly in the sport, is communication. How well you tell your concepts to a person who doesn't know zilch about medical sciences. And they fall for those misconceptions, because I see a lot of coaches exactly telling the same thing. If you're not tired, if you're not sweating, or t-shirt gila hona chahiye, you should see, even when Milkha Singh, you should train that bucket scene where, he removes his vest and, fills up the bucket with sweat and then continues running, and then everything's history. These are just few athletes here and there. For every one Milkha Singh, there might be 1,000 Milkha Singhs who might have been injured or might not even made it. What about them? Anyway, so head coach, assistant coach, video analyst, and the video analysts I've put there because they tell us a lot of data. I started my career with the Indian hockey team just before the Tokyo Olympics, both the men and women, and a lot of injuries we picked up by sitting with a video analyst. Non-contact ACL injuries. Most typical ones. Kya hua nahi sab, wo gir gaya aise. Aise kaise girega yaar? There would be some, and these you're talking about elite runners. They know how to run with the stick, with the ball, without the stick. So, without the ball, sorry. So a lot of information can be gathered through them. So when we talk about history taking, it's not just them, as opposed to even the masseurs. When they go for the massages, these masseurs have been there for the team for the past 5 years, 10 years, 15 years. Srijesh, the legendary goalkeeper, came with the calf pain. So then the masseur told me, "Aaj ye thoda sa muscle tight lag raha tha mujhe." Very good data, very important history. And again, the sports science team, the back-end team, which works, physiologists, strength and conditioning, biomechanics, the reason I've put the asterisks is that very few you can see them on the field of play. You can see the others, physio, yes. Doctors, sometimes yes, no. I've been lucky to be there sometimes. Anthropometry, but all of them contribute to this playing 11 or the playing 5 in basketball or in hockey or wherever they are. First, it's important to define the population who you're working with. And again, I've worked in a very small town called Bengaluru with a varied number of people. So it depends on who comes to me and what advice do I give them. Can't have a one size fits all approach for school competitions, you junior, masters, and every some or the other thing is happening in a city like Bangalore. And the last thing I want to do is that when you talk about history taking in the third term, we talk about history taking in a very, very clinical way in MBBS. Unfortunately, that's the trend even till date. But in sport, there's a very relevant history which needs to be taken, is training load. Unseen, unheard. So if an athlete comes to injured, when I was interning in my orthopedic postings, they again just spoke about, nobody asked about bone density. Is there any family history? They just ruled it out that if there's nothing like a family history of fractures, and that's changing. So when did you start the sport? At what intensity you started the sport? Who is your trainer? What kind of sport do you play other than your primary sport? So for all I have cricketers who, like, in the meantime, they play volleyball. And that time they've had an ankle sprain because the body is not used to that kind of jumps. Although they're doing it recreationally and they're elite players in one sport, but recreationally, just to take a break from that sport, they'll go on doing something. Classic example is Yuvraj Singh missing the World Cup. Glenn McGrath missing the Ashes. He was playing football just before the Ashes test match. Twisted his ankle, out for 3 test matches. So again, whether the sport is contact, non-contact, time bound or non-time bound, individual or team, outdoor, indoor, these are the factors that affect the human body and how they compete. And of course, my favorite is that working with para athletes, it's a whole new dimension altogether, which really challenges every word that you've studied till date. So what is sports festival? There are 2 teams. This is the Indian hockey team, the women's team at Sports Authority of India, Bangalore. Can anybody tell me what is going on there? What does it look like? What are the girls doing? What test would that be? It's called a yo-yo test. I don't know whether you've heard of it, but it's a very common parameter that is used across sport. And then there's this young fast bowler. I came across him while we were at the trials of the Rajasthan Royals camp last year. And pretty good fast bowler, but he didn't make the national team ever. And I don't know where he's right now, but he told a very interesting thing. When do you run a whole 2 kilometer consistently on a cricket field? When they say you can't play, you're not only coming at my cricketing skills, damn good bowler. But then he was struck out because he couldn't complete those 2 km in that particular time limit given by the South African Cricket Board. So again, it begs to differ that how much should be tested and how much should not be, and most importantly, how much is relevant. So just a differentiation between the 2 sports, which happen to be the 2 most popular sports in the country, if I may say. For cricket, again, intermittent, high intensity sport. For days in test cricket, you're just standing, third man, batsman is scoring a double century, you don't do anything. But then suddenly you have T20 cricket, you just keep hitting a batsman again there. So cricket is one sport where if a cricketer walks in right now, just by his body type, it's very difficult to say which sport he's in as compared to, say, basketball, hockey, football, athletics. You know a thrower from a jumper, from a runner. But in cricket, it's open for all. You have a Yuzvendra Chahal, who might be, 55 kilos? And you have a Rakeem Cornwell, almost 150 kilos. That's the beauty of cricket, or the lack of it, right? So again, the varied roles in cricket depends on whether you're batting, bowling, or wicket keeping. To give you certain insights is that we track the number of throws by each player, whether he's a bowler or a batsman. So inevitably, when a bowler goes back to field, you might have seen the bowler always on the boundary line. The reason is that his throwing arm is much better. He's better trained. He's adapted towards it. Very rarely you will find a spinner who's on the boundary line. Jadeja is an exception. because he's good throughout. What sir was telling about being naturally gifted, that boy is that. Coming to hockey again, aerobic capacity, anaerobic, it's a burst. 50, 60 minutes. I say 50, 60 minutes because it's rolling sub that is started in hockey. An average hockey player spends around 40, 45 minutes. Keeper might play the entire game, but he moves that much more less. Midfielder, forward, defender keeps rolling. And they have bursts which goes up to almost 30 to 35 kmph. That, and they have to decelerate as quickly as they accelerate, because it's the ball that is guiding their run of play. And the common injuries, again, it's a very basic thing that I've made is that your hamstring pulls, groin injury, ACLs. But in cricket, you see a more of a lower back injury, and that is very specific to fast bowlers, not spinners, not batsmen. And finger contusion, of course, the ball is hard. If anyone has seen how hard the cricket ball flies, it's like a missile. With the boundaries are so short these days, bats are so well. So what all the commentators say, most of them have been players. But once you go on the pitch and you see them, it's really hard. It's not the gully cricket that you play, right? The focus areas, again, the aerobic, anaerobic condition, drills, agility. So you saw the yo-yo test. Now, again, the yo-yo test is out of the cricketing system for some time. There's a new trainer, new head, so he'll get the winged test. And it's all very data-based. It's all very individual-driven in cricket. But in hockey, it is more of a team sport. Cricket, namesake, is a team sport. Because once there are two batsmen playing there, the other 9 are absolutely useless. You'll never find that in hockey. All 11 are equally there, intensely, fiercely playing. Okay? And their roles are much sought after. A defender in hockey is generally one who takes the penalty strike or the penalty stroke. Right? In cricket, you might not have a batsman coming in 9 down or a bowler coming 1 down, unless, of course, a night watchman, that's an exception. Again, what is load management? AC ratio, anyone? What is the AC ratio? Acute chronic workload ratio. Very important to define the eventual percentage of injury. Anything more than 2.1, we drag them back. That is, acute ratio is how much of workload has been done by that particular player in a day. And chronic is in a week. So we get a chart, we get a graph, and then we monitor each and every rep or set that particular player has done in that week, and then the entire month. If it's a game, great. If there are no games, off-season training, then they get a ratio. So that's how we monitor in hockey, but that's not done in cricket because it's not required in cricket. Right? Then, of course, we have some fancy gizmos. The GPS trackers, you might see those West Catapult, what's been used, force plates, and for monitoring data. And of course, the heart rate monitors, you have Polar, you have some smart watches, you have the hoop data that is calculated even for sleep. Then again, the training demands is some focusing coming out of hockey and out of cricket to after taking 2 team sports. This is like individual sports. For athletics, what sir was saying, short and long distance, you'll know when a 100-meter runner walks in, you'll know that he's 100 meters. But when a marathoner walks in, you'll know that he's marathon. Both are incapable of switching their respective events. Then again, for throwing events, you need a lot of more rotational strength, explosive hammer throw, discus, shot put, javelin. So you see the body types are very different. Even a javelin thrower might have a similar physique to 100, 200, but then his core strength is a lot more heavier. Shot put, even more heavier, because he doesn't have that longish run up. He just has that quick turn to take, right? Then again, sprinters, the acceleration drills are different, resistance sprints, start mechanics. So, I mean, it's calculated to the second, where from the start, how good is your reaction time? A shot put thrower might not have a great reaction time, but 100-meter from the start, when he's off the blocks, the time taken from when he perceives the sound of the gunshot and then when he takes it, all your neuromuscular junction, your neural activity, everything needs to be on point to save that 1 nanosecond. Actually, it's a very interesting stat. This year's Olympics in Paris, all the 8, 10 runners in 100 meters, had better timing than the 1992 gold medal winner of the 100 meters. I mean, that is something that is absolutely an insane data, right? So that much progress, and the difference will be what? Maybe 0.1 of a nanosecond or something like that. So these are some of the tests that we do in the field of sports science. The Wingate for anaerobic mobility, flexibility, stretch. And most of these, let me tell you, is quite inexpensive and can be implemented in our day-to-day practice also for recreational athletes and for the lay population. You don't need hi-fi gizmos for it. I think maybe, of course, leave the force plate aside or a proper VO2 max machine unless you're an institute. The others are quite easy. Like, for example, the muscular endurance, a push-up test, a pull-up test, a sit-up test. We can do it right now, right? So this is a very nice and a very, I can have the whole lecture on this one slide, actually, the talk. So you can see, you can place your athlete wherever it is, more the endurance, depending on the strength and speed. I discussed these two sports that I work with. So hockey is more on the endurance side and more little on the strength and speed. Cricket is somewhere there. You have the 100-meter runner. I mean, it's just a very basic pictorial depiction of what I want to convey. This again is to who comes to me and how do we treat them, how do we train them. Many say it's the human body. What will be so much that will change in the anatomy or the physiology, but actually this is the recreational morning runs in Kantirva which goes on, and that's the Indian hockey team which was training for the Paris Olympics. So both the approaches are widely different. The basics might remain the same, but intensity varies hugely. So what's the difference? The goal for elite, it's performance, performance, performance. They don't care about anything else. You know, you try to talk sense to the coach, but then according to you, it makes sense, but according to the coach and play, it doesn't make sense. So many times you put your body in line for the country, and why was X player rested for one particular match? Or why did now picture this last World Cup game, you're in the final, it's hockey, this last minute, a team is 1-2 down, somebody has cramps. So ideal medical knowledge will tell you, please take him out. But then somebody out there will be like, "Nahi, yaar, we need him." After that, even if he breaks a leg, I don't care. And this even the athlete tells. So it's it's a dilemma. It's not that straightforward. It's very good to read in textbooks that it's always health first, health first, health first. But sometimes, unfortunately or fortunately, again, it is not always health first. It can be sport first because there's so much riding on sport. The program, again, for elite sport is periodization, data-driven. For us, people like you and me, it's just basic progression. You know, you don't need to go on and fight to achieve the stars out there because no one is coming to give you a medal. Your medal will be longevity and good health. Recovery, daily strategic tracking, minimal focus. And the best mode of recovery is sleep. I mean, it's easy, right? You just have to sleep. Monitoring, we have the AMS, that's the athlete monitoring system. There's a whole entourage of people looking after these elite athletes, right from your respiratory rate, your heart rate, beats per minute, VO2 max, you name it, and you have everything recorded year on year with these people. But what do we have? Some of us just the smartwatch or the best device all clinical doctors, all the doctors will have is you can check your own pulse. And that's, I think, good enough. Last, the staff is obviously, you have the multidisciplinary team for elite, but as sir mentioned, you just have that one trainer with a hell lot of knowledge that he thinks he or she thinks he knows everything about it. And the major challenge is to bust those myths and misconceptions of they have a visually looking good body out there. That's why people fall for that and nahi mujhe aapki tarah banna hai. But more often than not, it's not the right approach. And more importantly, it might not be sustainable. Then the incentive, of course, performance and livelihood, money there. Here, again, it's just health. I wouldn't say just health. Health is a big word. But anyway, the intensity proportional to risk injury. And here, focus on consistency. No matter how little, please do something regularly, daily. And the return to play. I'll give a small example coming to a surgical picture. With any athlete, we're always fighting against time. In elite sport, the give or take time in elite space is not more than 7 to 10 years. Doctors, lawyers, journalists, everyone can have a 40, 50-year career, but an athlete will not have more than a decade on an average. That also, I think I'm saying it more. So the challenge remains, if there's an injury, if there's a surgical case, if something untoward is happening, he's losing time. And if you're losing time, you're losing a crucial period of your career. So it's imperative that for an athlete, we take them through that danger zone. And that's where game changes. Whereas people like you and me, lay people, may not even want to go to that danger zone. ACL, torn, repaired, go easy, come back on your feet after 1 year. It's absolutely fine. But an athlete can't take that much time, unfortunately. 8 to 9 months is the deadline given to even us, that we want this guy for the next Asian Games now. Now you decide how you'll work with him or her, right? Again, the sports text dot what we use, it's there available online, and some of them we use it on a regular basis. Since it's the physiology predominant conference, so I thought this pick will make sense. How it started, this is our lab I'm a consultant at Ramaya Medical College. So that's we use quite a bit of testing for marathoners. And that's the portable one. I think Inspire Institute is using it in Vijayanagara. So yeah, how much is too much? For the athletes, I would say nothing is enough. Just that how much we want to give them, that's our call. Get as much as possible, track them all, and then see how it goes. Any top leagues in the world, be it the English Premier League there, Big Bash in Australia, IPL in India, and Caribbean leagues in the Caribbean, everyone is so heavily investing in data. We can't accept it. We have to imbibe it. But then again, we would never know how much is too much. But for the recreational population, it's not necessary. For the recreational population, this is what is necessary, right? And the first speaker for this session with the Fit India movement, the Khelo India, and so many other initiatives by the government, they're all marketed and packaged in different names, and the bottom line name the same. Keep moving. How you move, we can guide you, but no matter how many schemes come up, whatever said and done, unless and until it comes from within, nothing is going to happen. Again, yeah, so just exercise snacks can boost your muscle growth. It's just another study. I have two studies which I really liked, was this and the next slide, which is there. So the slightest of things which even the previous speaker mentioned was that whatever little that you do has extremely good positive repercussions. Don't think that ये करने से कुछ नहीं होगा, बहुत कुछ हो सकता है. Okay? And this one, the age of 30, there's growth. You have to do something, your muscle hypertrophy easily. After that, you just have to do something to keep it static. So if you're not doing it, your sarcopenia is kicking in. And it's all about muscle, muscle, and muscle. Again, recovery, as I told you, it's very easy. After this, before you all start recovering here itself, it's just sleep, and good quality of sleep. It's nice to be preachy and say, we work duties for 18 hours ka duty kiya, 19 hours. I'm from the relatively newer age of doctors. So I'm very happy with my 8 hours of sleep, and I make no bones of it. Nutrition, hydration, and then comes the massages and all. So it has a lot of psychological effects. Of what I've seen with my athletes is that they promote some fancy gizmos in terms of massages and all that, but that is just maybe 2 to 5% of the total recovery that is required. Of course, then what about this population? Just food for thought for you all. This is not my topic for the day, but it means it's so close to my heart that I've been the chief medical officer at the Tokyo Paralympics and the Paris Paralympics, and we are training for the next Para-Asian Games. Just spare a thought. And how does this mechanics work? I would request all you professionals to, dig deep, read about them, and see if you can help them out wherever they are. Thank you so much.
Thank you so much, Dr. Ramya Kagali. Thank you so much for your presentation. Because of the lack of time, we can have a couple of quick questions. Yes, sir. Sir.
What about this phosphocreatine? There is a lot of people do ask me also. So I would take your advice on that.
So again, it's from where you get the source. If you're getting it in your diet, and if it is specific to your sport, please continue. If the option is not available, then we have to get it. Again, For elite, because the margins are so less, we give. My youngest player, and as you also mentioned about going to the gym, and it's very similar as going to the gym. Now in pediatric age group, going to the gym means it's such a taboo. Like it's not about going to the gym or not going to, it's about what you do in the gym. Similarly, phosphocreatine, no, it's not about from where you're getting. It's how much are you getting. If you're already getting it your meal, fair enough, don't touch. And that's why the assessments are so important.
Yeah, so because my advice generally has been that unless you're doing elitist kind of sports or
Correct.
very
That's-
high powerlifting,
Yeah.
you perhaps don't need it. Well, I thought you, since you are
Absolutely.
in the field, it will be
Absolutely.
confirm what we thought.
Absolutely. So, Sim, my youngest patient is an 8-year-old swimmer, already training for the Swiss Canal, and then later she has the dream of also getting into active sport in the Olympics in Karnataka. And she comes to the center. She lifts weights. Absolutely no harm whatsoever. It's how you lift, how much you lift, how well you recover. So the science is going into a lot of depth and not making blanket statements like, children should not go to the gym, your height won't increase. It's absolutely ridiculous, you know. And for the first time worldwide, you might have seen the number of overweight children have outnumbered the underweight children. This I was just reading in the news. I've not verified this news. I just saw in my InShots, which I was saying. So, I mean, that is scary. Yeah.
Home discussion, because my daughter loves to run, so she runs around, and then she wanted to increase her speed in sprinting. So she started reading, say, can I start doing weight training? So wife was absolutely against it.
Absolutely.
Yeah. Bilkul nahi. Till the time you mature, I'm saying it's okay.
It's okay.
Please
Absolutely.
go. So, and this is a common thing which is now occurring for children. So I think your input on that you can exercise, you can do a bit of, is that correct?
100%. 100%.
Dr. K.K. Deepak sir wanted to ask questions, sir.
So,
Question, the physiologist role in sports sciences, what India needs a data set. We have not maintained our data. The devices, instrument, most of them are imported. So we have not made our own device. If you don't have devices, you don't have data. And otherwise, it becomes so difficult. We recently entered MOU with SAI and IIT Delhi. Now at IIT Delhi, we are having our own data set, and then we apply AI.
the
Because that comes
NCSSR
later.
is doing a good job, I guess.
So and now we have entered another field like parathletes, how we can increase their performance by having instrumentation. So all kind of IMUs and other things that we're trying to. So as a physiologist, you should pitch into data collection. You are the best person, you can collect the data set. But the biomechanics, the nutrition, they are all their psychologists. But we have not played a role. All the best.
Yes, madam, last question. Yes, madam, please, go ahead.
I have basic questions like several times in India, while selecting the sports, there is no parameter to assess their muscle strength. So is there any data which is available to say that uh, sports injury is due to the nutritional deficiency, and that is the reason, even though they are selected, shortlisted for the national team, and they get injured and they're not able to perform well, is it one of the reason for we are not getting into the main front of any of the major, like in Olympics or anything? So we're not in the top lead.
The question is good, but the answer won't be that simple. So what I would say is that yes, the lack of data definitely persists. But things are changing. I mean, and India is a country which will crawl. And the only reason I say so because we are still not a sporting nation. I also say that we are not even a cricketing nation. People will be like, oh, what nonsense. I'm like, no, we see IPL. If we are a cricketing nation, we'll also be seeing Ranji Trophy, we'll also be seeing Duleep Trophy, we'll also be seeing India West Indies Test match that's going on right now. So by far, we are not a sporty nation. When you're talking about a sporty nation, it's like, even when you get a breakout here for students, maybe MBBS or whatever it is, they might just go for a quick cycle. Or in the morning, they can come, go for a quick workout, and then they come. Because that's what people do abroad. I did my PG in New Zealand. And little break, let's go for a cycle. So what they were saying, nothing goes waste. So that whole mindset is there, not to become an elite athlete, but just to stay fit. Once that is taken care, that's the first step of playing sport and playing athlete. Now what we think is that yaar sabko abhi now mujhe IPL khelna kyun hai, paise mil rahe wahan pe. That is not going to take you anywhere. And same thing, the nutritional deficiency, everything is just like a cascade of events as such. And what goal they've started also, that also changes. Those who come, they will come with a spurt of brilliance that, 100 meters I ran this much and all that. Once they start coming to the system, then they know. Main thing is that going to number one, yes, you can go, but sustaining there is a challenge. So nutrition definitely is a very, very important cog in that wheel. And there is no research, unfortunately. We are banking on you for that.
Thank you. Thank you so much, Dr. Amya. Thank you so much. And with this, we will conclude this session.
I request the chairpersons to provide the certificate of appreciation with a memento to the resource person. I now request Dr. Lakshmi T. Rao, Professor and Head of Department of Neurophysiology, NIMHANS, Bangalore, to kindly provide a certificate of appreciation to our chairpersons.
Non può,